afternoon. So I'm Ed Conway, in case you haven't met, and there are many faces that I really haven't met. Welcome to our annual CBR Research Day. This is a terrific afternoon to increase stress and the stress levels of our summer students. It's sort of the culmination of stress, but they're going to wow us this afternoon, no doubt. First of all, I'd like to start by expressing thanks for the people and the organizations that enabled this first through financial support from CSL Behring, from Pfizer, from Grifols, from BioVeritiv, New England Biolabs, Bayer and the Bayer UBC Bleeding Disorders Collaboratory, the Canadian Blood Services, Octapharma, Mako Pharma, Stego, Alexion, as well as the Sheldon Neyman and Linda Vickers Hematology Endowment Fund and the Neil Mackenzie Memorial Fund. So all of these organizations and funders have provided the money so that we can hire these starving students and put them into our labs and clinics, and to work for us effectively. We really do appreciate this opportunity. It's a really unique program that is really, I think, the largest on campus and the most comprehensive. And I think most of the students have had a great summer, mostly because of the hard work of Anna, who's not here right now. She should be delivering imminently. Hannah and Anna and Mira and Julie, who many of you have met. So we really owe them a thanks. I don't know if any of them are in the room right now. But when you do see them, please thank them, because they are really the ones who did all the work. And then I would make sure to thank all the research technicians, the postdocs, the PhD students, the master students, and occasionally the PIs and the research associates who have supervised these students in achieving what they're going to describe today. Now, our program, it's a little different. So first of all, the student presentations will be 3 minutes exactly. And if you go over by 30 seconds, you will be pulled off. At the end of and after each presentation, all of the students have score sheets, and they will score all the speakers, including yourself, so I'd recommend that you give yourself a good score, okay? These are anonymous, so we won't know, and then we'll collect them, and there will be an award. So the presentations are, the award is really by your peers, all right? So please do, and then I'll collect those at the very end of all the presentations. After which there will be a Neil Mackenzie Mentorship Award, and you'll hear a little more about that by Dieter Brahma, who will present that. Now, our keynote has changed. Last minute this morning, I had a 5 o'clock in the morning email. Occasionally I answer those. And our keynote speaker, Farah Alibay, who's an engineer, astronaut-like kind of person who did her PhD in planetary exploration. It's sort of ironic that her airplane was grounded, and Air Canada wasn't flying that plane. It was canceled, and she really could not get here in time. So she expresses her severe apologies, but we will get her next year. But in the meantime, we have somebody to fill in, and this isn't just a fill-inner. You'll be very pleased by the speaker, Jamie Matthews, and I'll introduce him later. He's not here yet. He's sort of interviewing media, I think, about the solar eclipse. After that, there will be an open bar immediately, so you can walk out, and there'll be drinks as you walk out the door, and the students will need that. And then there'll be posters and poster judging, and a barbecue dinner, and awards, and all kinds of thanks. And that's Jamie Matthews, just to show you what you're in for. Okay, this was one of his regalia. Now, the student presentations, all right? So, a few things to remember. Do not look back. This is being videoed, all right? And you don't really want to have a 3-minute picture of your right ear, okay? So don't look back. There's a presentation in front of you with this giant pointer, okay? So if you need to use it, which you probably don't, it's there, okay? That's one. Target is 3 minutes. At 3:30, you'll be pulled off. That's when the hook comes, and you will be pulled off the stage. And then, right after that, the scoring. So please do remember to score, and in the order. Okay, check the name, but I think we've put it in the order of the speakers so as not to be too confusing for your minds. Okay, so without any further ado, okay, no walking, by the way. That's what the chairs are here for. You can't move, so stay here, otherwise you'll be out of earshot. Okay, Alvin.
So hello everyone, my name is Alvin Wong and I am part of the Magnanti lab. And so today I'll be talking about a protein that I've been studying this summer, and it's called podokelixin. And podokelixin is shown here, where it localizes actin to the apical membrane of podocytes. And podocytes are important because they have something called foot processes, as you can see here, that allows for filtration of the blood to make urine. And as you can see here, without podokelixin, there is no foot processes formed, and this is what causes end-stage renal disease. And so, in previous studies, they did a total knockout of podokelixin in the entire body. And so for us, we wanted to delete podokelixin specifically in podocytes. And so we used a mice model under the Cree-LOX-P system, where the Cree cassette is under a podocyte-specific promoter. And what we found is while our wild-type and het mice, they all survived past 8 weeks, our knockout mice all died between 3 to 7 weeks postpartum, or after birth. And naturally, we wanted to discover why they died, and so we did a series of stains. So first, we did a Mason's trichrome stain, and as you can see, compared to the wild-type kidney on the left, and to the knockouts in the middle and on the right, you can see evidence of matrix protein deposition and also deterioration of kidney infrastructure. And this is indicative of renal disease. We also did a urine sample analysis using SCS-Page, where we are able to find that there is a presence of albumin in our knockout mice urine. And this is a clinical definition of proteinuria, or protein in urine. And we also did a DAB stain to prove that our model did indeed knock out podokelixin. And as you can see in the knockouts, in the peripheries where the podocytes are, you can see there's no podokelixin expression. Furthermore, we did a stain for P57, and this is a protein that's specifically expressed in podocyte nuclei only, so we can quantify the number of podocytes between the groups. And we found that between the wild-type and knockout groups, there is no significant difference between the two, which suggests that for our disease, it's not associated with a reduction in number of podocytes. We also did a stain for desmin, and desmin is generally overexpressed in injured kidneys, and we see this overexpression in our knockout mice, which suggests that there is morphological changes that's causing the kidney to become dysfunctional. And in conclusion, our podocyte-specific knockout of podokelixin does indeed cause end-stage renal disease, and we are interested in looking at a potential drug called BIS-T23, which is shown to promote actin-dependent diamine oligomerization and may have a role in actin localization. So we would like to see if we administer this drug into our knockout mice, will it ameliorate the disease phenotype and in the absence of podokelixin. Thank you very much.
So my name is Shannon Sui from the overall lab. Proteases are largely defined by the function and range of the protease of the substrates, and they are all interconnected in a complex protease web as seen on the right-hand side. So the focus of my project is plasmin and tissue plasminogen activator. Plasmin is a major protease in blood that cleaves fibrin clots, and TPA activates plasminogen into plasmin. So sometimes it is used to treat stroke. However, treatment with TPA may cause hemorrhaging. So the aim of my project is to use proteomics to discover new substrate of plasmin and TPA, and also elucidate their role in the plasminogen system. A human breast cancer cell line and a human umbilical vein endothelial cell line were used. The secretome and membrane-enriched fraction were collected and treated with buffer, plasmin, or TPA. Terminal amine isotopic labeling of substrate, also known as TAILS, is a technique that allows identification of substrates in complex samples. So buffer treated or protease treated samples are isotopically labeled with formaldehyde, which blocks lysine residues, natural N-termini and the new N-termini generated by the protease. So this one will stop the binding to the polymer. These peptides are then recovered by centrifugation and then analyzed by MSMS. And then we compare the peptide label ratio to find out which is the potential substrates. Currently, there are only 52 substrate of plasmid list of MEROPs, but using TAILS, we find 370 unreported substrates of plasmid in breast cancer cells and endothelial cells. But these are only potential substrates, so we need to validate by visualizing them on the SDS-PAGE gels. One of the substrates we found of plasmid is fermentin. As we can see from these SDS-PAGE gels, fermentin is cleaved by plasmid at different molar ratio. And when plasminogen is added to raw 264.4.7 cells, the cellular TPA will activate it into plasmid, which cleaves the fermentin that is added to the cell medium. Fermentin can also induce chemotaxis in THP1 cells, but when it is cleaved with plasmid, fermentin loses its ability as a chemoattractant. Overall, TAILS is a very useful and powerful technique to find new substrate of proteases, but each step needs modification for different proteases. And the second part of the project, which is doing TAILS with TPA, is still in progress.
Okay, so hello, my name is David. I'm from the Conway lab, and wait, can everybody see me okay? Yeah? Okay, because that means you don't have macular degeneration, which is the leading cause of blindness in the elderly. This is characterized by neovascularization in the retina, which is kind of like the formation of a leaky vessel in the back of the eye. So this is due to damage of a membrane causing angiogenesis, influx of leukocytes, and inflammatory damage. Now, thrombomodulin is a widely expressed transmembrane protein with known anti-inflammatory effects, especially its lectin domain, which is why we thought it might be a potential therapeutic for treating AMD. So I'm going to say thrombomodulin a lot, so just keep in mind I'm referring specifically to the lectin domain. First things we did was use an in vivo AMD model, where the same type of damage to the membrane was induced by lasering, and then treated with different concentrations of thrombomodulin. As you can see, compared to the no treatment on the left, lesion volume was decreased with the treatment of thrombomodulin in a dosage-dependent manner. Next was trying to figure out how this was exhibiting this protective effect. First thing we did was run a real-time on known molecular markers of inflammation. And as you can see, human retinal cells treated with thrombomodulin showed a decreased expression level of intercellular adhesion molecules, which are important for the adhesion and transmigration of white blood cells during inflammation. And to see this more clearly, we did something called an adhesion assay, where thrombomodulin-treated epithelial cells showed decreased levels of adhesion of white blood cells. And for transmigration assay, we treated endothelial monolayer with thrombomodulin, and we saw a decreased level of leukocytes passing through that monolayer. Next, we ran a real-time against proangiogenic factors known to be associated with macular degeneration. And again, human retinal cells treated with thrombomodulin showed decreased expression of something called VEGF, or vascular endothelial growth factors. So to visualize this, we did something called a tube formation assay, where cells are allowed to form tubes to mimic new vessel formation. So even in the presence of a proangiogenic factor, thrombomodulin is quite effective in decreasing or suppressing tube formation. So what can we know from all this? Thrombomodulin lectin domain protects against AMD in mice, and the mechanism by which it does this is suppression of inflammation and angiogenesis. And the lectin domain of thrombomodulin is a potential therapeutic for a disease that is not yet adequately treated for. Thank you.
Hi guys, my name is Chelsea, and this summer I was working at the Genome Sciences Center at the Carson Lab. So, reproducible research is something that hopefully we're all familiar with. With any study or claim, having reproducible research allows for data analysis to be successfully repeated and verified. Recently, whole genome bisulfate sequencing has been successfully done on single cells, allowing for a way to closely analyze cell-to-cell heterogeneity, crucial in stem or cancer cells. While some papers have been published on the technique, no standardized method has resulted in slight variations between them all. This is where SCREW comes in, single-cell reproducible epigenetic workflow, something I was working on this summer under the supervision of Kieran O'Neill. SCREW is a series of CWL and Dockerized tools that allow researchers to create reproducible sequencing analyses. CWL, or Common Workflow Language, offers a way to create and represent bioinformatic workflows, while Docker gives the ability to reproduce the exact environment the initial analysis was created in. This is the master workflow that is kind of envisioned right now for SCREW, with the highlighted sections being completed. So, this summer, when I came in this summer, all the basic analysis tools were already in place, and I was working on updating the workflow by connecting the pre-processing and clustering tools. So pre-processing is kind of a preliminary step to kind of sort the data and format it in the way we want. So these three first data formats are kind of the different formats that were published, and having completely different formats is really hard to work with. So pre-processing, the pre-processing tool in SCREW kind of sets it to the format below, which is the one that's used by all the internal standards in SCREW. So the clustering tool is one that includes the computing of pairwise distance based on CpG methylation and the plotting of a heat map based on that distance matrix. Using the Farlick study, I was able to apply our workflow onto a real-world data set. The Farlick study was an analysis done on human stem and progenitor cell data, with the data gathered using single-cell sequencing. The data was run through a complete SCREW workflow, running through all the pre-processing and clustering steps I mentioned earlier. In the clustering section, I was able to specify a bed file for the workflow, which contained locations of active enhancer sites in CD34 bone marrow, which filtered the CpG sites more likely to be of regulatory importance. With CWL, SCREW is easily adapted and altered, so researchers can add and take away tools that they want in their analyses. This makes it clear to see how analysis is run with the execution of just a simple script. With simple template workflow, a standardized method for single-cell DNA methylation analysis can be implemented and available to reproduce results. Thank you.
Hi everyone, my name is Caleb Ritchie, and this past summer I've had the opportunity of working in Hancock Lab on a bacteria called Pseudomonas aeruginosa, and I was looking at peptide treatments. A bit of a background before I get into my project. Pseudomonas aeruginosa is a opportunistic and very persistent bacterium which causes very chronic and hard to treat infections once they are established. This is largely due to their ability to form biofilms. Now, what is a biofilm? You've probably learned about them if you've taken microbiology, but it's more than just a cluster of cells. It's a complex community growth mode which can confer massively increased antibiotic resistance. Now, because of this, development of new biofilm targeting drugs are urgently needed to reduce the burden on the healthcare system caused by this bacterium and others. One option is anti-biofilm peptides, which have shown promise in the Hancock Lab at eradicating and inhibiting biofilms. So my goal this summer was to identify mutants from a large PA14 transposon library that are differentially affected to these peptides compared to the wild type to try and understand the peptide mechanism. So this involved growing biofilms for 24 hours, treating them with peptide, and measuring with crystal violet staining the change or the differences in biofilm eradication. I then analyzed the results and grouped mutants as either resistant to peptide or hypersusceptible compared to the wild type. Some further testing that I did was the minimum concentration of peptide that was required to inhibit, also adherence to plastic, and I looked with some confocal microscopy to see if I could visualize these differences. So the screen yielded several previously described genes, as well as many genes that I was able to group as resistant or hypersusceptible to at least one peptide. Also shown is the results of the minimum concentration of peptide required to inhibit biofilms at 20, 50, and 80%, as well as adherence to plastic. The images shown is of the CLIP-A mutant in PA14, which showed resistance in all three areas shown on the screen. This protein CLIP-A has actually been shown to bind to protein CLIP-P in E. coli, and that CLIP-P protein has also shown interesting biofilm resistance phenotypes, which could be related to the results with the CLIP-A mutant. So I believe that further study in this area could contribute to new and better biofilm targeting drugs and reduce the weight and the burden on the healthcare system caused by antibiotic resistance with this bacteria. Thank you.
Hi, my name is Leo from Hancock Lab. There's a plethora number of pathogenic bacteria, and in fact, a bacterial infection, once thought a resolved problem, is a re-emerging it's a global threat in both clinical and community setting. Our best solution is employment of antibiotics. However, as you may know, bacteria can have acquired intrinsic or even adaptive resistance against these antibiotics. These could arise from misuse or even proper use of antibiotics. And take an example of methicillin. It was developed in 1960, but the resistance was found in 1961. Daptomycin was approved 20 years after its discovery, but the resistance was observed in 2 years after its release. So it's a cycle of tag and tagging back. But as you may know, bacteria or organisms are much better at chemistry and biochemistry than chemists and biochemists themselves. So there's a high demand of alternative antibiotics. Some of the way that Hancock Lab addressed this problem is by understanding how the resistance could emerge or directing the therapeutic target to hosts from the pathogens, or seeking new molecules for therapeutic activities, such as radicals, peptides, and lipids that I'm going to talk about. The summary of our projects is that the engineered liposomes, which are spherical phospholipid bilayer molecules, can sequester bacterial exotoxins. And then this is the scheme that I have, and I'll talk to you step by step. So first, we grow the bacteria and then sterilize the supernatants, which we think the toxins are in, but there's no bacteria. We give that to the cells, and you can see by both microscopy and in biochemical assays that these are all dying. But if you give a liposome, a sphingomyelin or cluster of sphingomyelin liposomes to these with the supernatants, you can actually see that cells are protected. So what we can do next is that we can isolate these liposomes, and then we can do a mass spec analysis, and we see what kind of toxins are actually bound to this liposome, thus protecting these cells. And then after that, we can take the data from the mass spec and then screen the mutants that we found the proteins of in the mass spec data, and then we see that if there's a reduction of cytotoxicity, we can conclude that is a contributing factor of a toxic factor in that supernatant. So what was actually in the liposomes? I guess you have to come to my poster to see that. The take-home message for me is that the liposomes can sequester bacterial toxin and thus protecting human cells. However, that's only part of the picture. So eradicating bacteria as well as inhibiting bacterial growth is a very important aspect of clearance of infection. However, liposomes cannot do that. So the Hancock Lab have several approaches and several strategies to collectively combat with bacterial infection, and I'm just part of them.
Hi everyone, I'm Lana Mazik and I'm a student in the Conway Lab. Today I'm going to be talking to you about CD248 and venous thrombosis. As the name suggests, this occurs when clots form in the vein, usually in our legs, where here we see swelling and redness. Besides being common and widespread, venous thrombosis mostly affects people with obesity, cancers, and inflammatory diseases. Because of these things, it's easy for us to see the significance of this disorder. But where does CD248 play a role? Well, this protein is transmembrane glycoprotein expressed by cells such as vascular smooth muscle cells and fat cells. Using genetically modified mice, our lab found that CD248 knockout mice had smaller clots in comparison to wild type. So this finding inspired my research with the objective to understand the mechanism in which CD248 influences clotting. To do this, I utilized ROTEM. ROTEM is a well-established technique that gives us information of the kinetics of clotting, such as initiation, formation, and overall stability. As you can see above, CD248 knockout mice had delayed whole blood clotting and slower formation, though the overall strength was the same. So this finding made me want to look further into it and differentiate between blood cells and plasma. So we repeated the same experiment with plasma. Surprisingly, we saw no significant difference between the two genotypes. These two results suggest to us that something cellular might be affecting clotting. So where do we go from here? Well, the mechanism of CD248 with clotting is still widely unknown, but with this information we have hints. So we've started to look into different cell-mediated and plasma-mediated potential roles of CD248. We've started establishing triple stains to visualize tissue factor expression in respect to perivascular cells because this is the control center of clotting. In addition, we're starting in vitro co-culture experiments to look at different response between wild type and knockout cells and to see if maybe there's a difference in the cells themselves. In addition, we'll continue using ROTEM to look at the potential of soluble CD248 in deficient blood. Overall, we see that to understand CD248's mechanism will give insight in venous thrombosis and may lead us to new diagnostics and therapies. If you guys want any more information on this, you can come visit my poster. Thanks.
Hi there, I'm Paul Murray and I'm from the Stranatica laboratory. So we focus on crystallography of proteins to determine if they can stop antibiotic resistance. My project is on the AMPG protein and I was sent to find its crystal structure. In this case, I failed completely and did not find it. So the reason why we look at the AMPG protein is because it's responsible for recycling of the peptidoglycan layer, which is then, if it goes too much when we add antibiotics, it'll cause too much glucanac and anhydrous Murnac into the cell, and that will then start the AMP-C receptor to come off and then stop lactases. So to start off, I did a cell-free expression because AMPG does not like to be overexpressed in cells. Next, I went and did a nickel chelation to get rid of contaminants such as the transcription factors I used to make my protein express. And finally, I went off either a detergent screen in an X-ray crystallography, or I did a nanodisc method because of the aggregates I was seeing, and did on the EM. So in my detergent screens, I tried using what my collaborators had previously used in the Pseudomonas gene on my E. coli gene. So I used DDM, DMNG, and C12E8. However, I only saw large expression in DDM, and then the aggregates made it so I didn't get a good crystal hit. When I tried it in nanodisc expression, as you can see, I did get my protein in it, but it didn't look that great as there was a lot of aggregates of lipid and then protein itself. So my future directions, I could isolate a more pure sample for the electron microscope, or I could try a new method in LCP. That's it.
Hello everyone. So I'm going to tell you guys about what my boss made me do while he went on vacation so that I wouldn't get bored, and maybe so that I would learn something too a little bit. So the goal here was to develop a method for separating intact proteins, meaning whole undigested proteins, via chromatography. I did that by playing around with 4 parameters on the front end of the system, the UPLC, Ultra Performance Liquid Chromatography System. And the back end of my system, the actual mass spectrometer, basically was there to tell me how well I was doing. Well, doing well at what? The goal was to separate a simple mixture of 7 standard proteins that I made. And in mass spectrometry, good separation is separation that will allow you to reconstruct the mass of those proteins faithfully. So, under my best conditions, I have gotten some pretty good separation. The peaks aren't quite, there's some overlap, and those little noisy bits at the bottom are due to the intensity of the signal means there's a high noise ratio, so they're not actual, they're false positives. But there is a decent amount of separation that the peaks are visibly separate from each other compared to the negative control, pointer, the negative control where you can see that only insulin has separated. The final goal was to see if I could reconstruct the masses of these proteins, and I did. Up here is the control. These are individual runs of the proteins, each 15 minutes on our previous system, and this is all from my optimum run right here. So the masses are pretty much the same. If I were to try different things, I would want to try different packing material. The chromatography we use is reverse phase, which means it separates proteins based on their hydrophobicity. And with the kind of packing material I use, that's limited. And I'd also want to try a different column length, which can give you more separation power, or for shorter column length, you can have shorter, faster runs. What was the point of this? Like, has this been done before? Yes, people have separation protocols for intact proteins. The point really was for me to learn about what method development is like, and for me to improve the system that we're currently using, which has more potential than it's currently being used for.
Hi everyone, my name is Colleen Wong and I am a third-year medical student studying in Dublin. And this summer I've been working alongside Dr. Heather Leach. And so what we've been looking at is iron overload in lower IPSS risk patients with myelodysplastic syndrome receiving red blood cell transfusions and the relation to either infection and possible benefit from iron chelation therapy. So MDS are clonal bone marrow disorders in which ineffective hematopoiesis occurs, leading to peripheral blood cytopenias. And most MDS patients actually develop a significant anemia, which requires multiple red blood cell transfusions. And as we know, of course, red cells contain hemoglobin, which are rich in iron. So unfortunately, the body has no mechanism to excrete excess iron, and over time, this damages tissues and organs. So just on the right there, you can see a transfusion with excess iron depositing in the body. So microorganisms use iron for growth and replication, and data suggests that iron overload actually inhibits neutrophil function, with some studies suggesting a higher incidence of infections and infection-related mortality in transfused MDS patients. However, no studies have addressed the infectious risk in patients receiving iron chelation to offload iron, which is why we performed a retrospective analysis of infectious episodes in transfused MDS patients requiring antibiotics, antiviral, and antifungal medications, comparing the two groups together. So we started with 182 transfused MDS patients, and we excluded 44 for either not being transfused as lower risk, being higher risk, or receiving a disease-modifying therapy and or having inadequate information. And we were left with 138 patients who were lower risk and transfused. And so this is just the infection-related characteristics of transfused patients comparing iron chelation to non-iron chelation patients. And as you can see, number of patients with infections, infectious episodes, type of infection, number of patients hospitalized for infections, number of infectious deaths, and the neutrophil counts at first red blood cell transfusion as well as the first infection all had an insignificant p-value when comparing iron chelation to non-iron chelation therapy. And so down here we just have the different types of bacterial infections experienced, and that was because they were the majority of infections, with iron chelation patients in blue and non-iron chelation in red. And there were respiratory, urinary tract, skin, deep tissue, and other. And so what's interesting, well, first of all, this is the graph for first red blood cell transfusion to first infection. But what's interesting to point out, and despite the previous slide, is that time to first infection was significantly shorter in patients not receiving iron chelation therapy. And again, the p-value remained significant in a multivariate analysis. So in transfused MDS patients, even though the number and type of infections were similar between the two groups, and that despite similar neutrophil counts, that actually time to first infection was significantly longer in patients receiving iron chelation. So these results suggest transfusional iron overload may contribute to infection in MDS patients, and iron chelation may help to delay these infections. Thank you.
My name's Aman, and I am a summer student in Dr. Kizikadektu's lab. And this summer, I've been working on polymeric osmolytes for protein stabilization. I have a little joke there if you get it. And so our background is that proteins are specific and potent. And since the FDA approval of insulin just about 30 years ago, there's been a remarkable expansion in this field, with now over 130 FDA-approved protein drugs in clinical use today. But the problem is that they're chemically and physically unstable. And so what pharmaceutical companies do is use trehalose as an excipient to stabilize them. Trehalose, as you can see the structure here, is a non-reducing disaccharide which can naturally be found in yeast, fungi, and bacteria. But the problem is that it's expensive and laborious to synthesize. So my hypothesis that I've been working on is the synthetic polymer, which I'm going to be referring to as HP, with high hydration pockets would allow it to imitate the trehalose mechanism and act as an excipient for different proteins. So this is a little snippet of my work, and you can come to my poster to check out more of it, but I started off with the chemical denaturation of hemoglobin using guanidinium hydrochloride. And so with this, I measured the UV vis of hemoglobin at 405 nanometers, which is characteristic of the heme group. And so you can see in the light blue is trehalose, and in the dark blue is our polymer HP. And it has significantly greater excipient abilities than trehalose. And so it shows that our polymer did work better than trehalose, and we plan on expanding our research towards beta-galactosidase in lyophilization, agitation in lysozyme, and then therapeutic proteins such as insulin and growth factors. I also just like to take a minute to thank CBR Summer Fellowship for this opportunity. Thank you.
Hi everyone, I'm Gayatri from the Castrop Lab. Currently, few methods exist for delivering therapeutics to sites of vascular injury to control hemorrhage and cardiovascular disease. When an individual is injured, platelets are the first to arrive at the site to control the bleeding. In brief, these platelets become activated, causing them to aggregate and release their cargo in the form of soluble proteins and small molecules, or release microparticles containing proteins and nucleic acids for targeted delivery to cells in the wound site. The project that I was working on this summer attempts to exploit the platelets' inherent responses to injury to develop a method for targeted delivery of therapeutics to sites of vascular damage. More specifically, we're trying to transfect platelets with lipid nanoparticles, or LNPs, containing mRNA, so that when the platelets are activated, they release these microparticles for targeted delivery to the cells surrounding or in the wound site, or translate the mRNA that we have given them so that they can release the biologics into the wound site. Now, before I go into explaining my results, I'd like to tell you all one of my main takeaways from this summer, and that is that platelets are like little kids. Getting them to do what we want is not at all the easiest, no matter how well we treat them. So the first step in this project was to determine whether the uptake of these nanoparticles is even possible. And to do that, we incubated LNPs with platelets that were either activated or not activated, washed them, and then quantified the uptake using qPCR. And we found that the uptake, which is shown here on the y-axis, is enhanced with time on the x-axis and activation, the black bars. We were also interested in looking into whether other known platelet agonists also display a similar trend. And we found that it is true. At the 4-hour time point, all the samples that were activated with an agonist displayed increased uptake. To ensure that the LNPs were actually entering the platelets, we used fluorescence microscopy and found that the LNPs do in fact enter the platelets, as you can see here by the little yellow dots inside the red platelets. We're also interested in seeing whether or not the mRNA that we're delivering is getting translated by the platelets. And unfortunately, using FACS, we did not see an increase in the green fluorescence protein fluorescence, and therefore we concluded that there is no translation of the delivered mRNA. But here's the catch. Platelets are known to synthesize proteins from their own mRNA when they are activated. So then why are they not translating the mRNA that we have given them? All the results taken together and visits to the literature have led us to hypothesize that perhaps the mRNA in the LNPs after being taken up are in fact getting stuck in stress granules, which we think are formed when they are activated, which is what is preventing their translation. So to know more about our stress granule hypothesis and what we have done so far, you'll have to come and see my poster. Thank you.
Good afternoon, everyone. My name is Jordan, and I'm a student at Kelly Brown's lab where we study rheumatology at BC Children's Hospital. And today I want to tell you about a rheumatic disease that most of you have probably never heard of before, childhood chronic primary vasculitis, or CPV. And the reason most of you have probably never heard of it is because it's a rare disease, and the entire province of BC will only have 1 or 2 new cases of childhood CPV each year. It's characterized by inflammation of the blood vessels affecting the major organs of the body, including the heart, lungs, kidneys, and brain. And the scoring tools that clinicians use to assess disease activity in CPV, PVAS and PGA, they're imperfect. They work well when disease activity is high, but not so well when it's low. And the problem with this is, if a patient comes into clinic with severe headache, rash, fever, and numbness, you probably don't need to look at some numbers to tell you that something is wrong. But if disease activity is low and there are no obvious symptoms, then we need more sensitive measures to help find the best treatment for the patient. You see, treatments for CPV are quite intensive, and over or under treating disease can cause significant problems. So we need a more sensitive, preferably biological marker of disease to help guide those treatment decisions. Enter S100A12. This is a damage-associated protein made by neutrophils that tracks with disease activity in vasculitis, going from high activity at diagnosis to low activity following months of treatment, as you can see on the bottom right. This makes it potentially interesting for us as a drug target. So we wanted to test the hypothesis that S100A12 is an active participant in vascular inflammation. To do this, we and a team in Germany exposed umbilical vein and coronary artery endothelial cells directly to S100 or TNF-alpha or LPS as controls. We also did indirect stimulation where we took PBMCs or monocytes from healthy blood donors, stimulated those with S100 or LPS as a control, and then took those supernatants and used them to culture our endothelial cells. Activation was measured by looking at 3 different things: gene expression, surface markers, and secreted factors. And in short, S100 doesn't directly stimulate endothelial cells, but indirect stimulation with S100 seems to upregulate pro-inflammatory gene expression in our coronary artery cells, as you can see on the left. And by adding an IL-1 beta inhibitor to those monocyte supernatants that have been stimulated with S100, we can attenuate this increased gene expression as shown by the arrow on the right-hand side. This suggests to us that S100 may signal to endothelial cells indirectly through an IL-1 beta signaling pathway in monocytes. Now, we don't see any effect with our umbilical vein cells, so more work still needs to be done to confirm this. So in short, S100A12 is a sensitive biomarker of disease activity in childhood vasculitis. It doesn't directly stimulate endothelial cells, but may do so indirectly through an IL-1 beta signaling pathway in monocytes. If you want to learn more, come to my poster, please. Thanks, bye.
Okay, $3 million. That's how much I'm hoping my project can save Canadian Blood Services every year. I'm Quinn Matthews. I'm a summer student at the Divine Lab, and my project is titled Big Savings in Small Packages, Non-Destructive Quality Control Testing of Red Cell Concentrates. So red cell concentrates are key blood products. They're transfused. Patients suffer from acute trauma, undergoing surgery, and more. So thousands of these are used in Canada every single day. In fact, in 2016 alone, Canadian Blood Services produced almost 900,000 of these units. And not only is that a lot of blood, but it's a lot of money. At $350 per unit, we're looking at the hundreds of millions of dollar range per year. So I'm sure you'd be surprised if I were to tell you that 1% of these red cell units end up in the garbage. And this is because current regulations state that 1 in 100 bags produced need to be held for expiry at 42 days, the blood cells expire, and tested for quality control to make sure the blood is being produced properly and to make sure it's safe for transfusion. So that 1% translates to about 9,000 bags per year, which ends up being $3 million yearly. So our lab group saw this and we thought, that's a lot of money, let's try to save that and let's try to eliminate this waste. So we came up with the idea of taking a small aliquot of blood from the red cell units during production and storing that small aliquot in a miniature bag. This miniature bag would be used for quality control testing, which would free up the red cell concentrate for actual transfusion. So we avoid this entire wastage of blood and money. But for this to be feasible, we had to make sure that the blood quality within the mini bag closely mimicked the blood quality within the actual red cell unit itself. Otherwise, the quality assurance is useless. So we designed 3 different bag types, and we filled each with 4 volumes, ranging from not very full to quite full within the bag. And we held these until day 42 and did the testing that Canadian Blood Services does. So we looked at common in vitro red cell quality markers like hemolysis, extracellular potassium, microvesicles, pH, and more. I can tell you all about this on my poster. And then we looked at the difference between the quality within the mini bags and the quality in the red cell unit itself. And we looked for the smallest difference, so the smallest delta, and that gives an idea of the best bag size. So here's a quick look at our results. Let's focus right now on the hemolysis graph here. So these are four quality markers that are commonly used for red cells: hemolysis, microvesicles, extracellular potassium, and pH. So I've graphed the delta here, so the higher the column is, the larger the difference between the mini bag and the red cell unit, which means that it's a not good choice, right? We want a smaller delta. So the blue, the red, and the green columns represent the different bag sizes. And as we move from left to right, the fill volume increases. So the general trend that we can see here is that as we increase the fill volume in all of the bags, the delta shrinks. So the quality in the mini bag becomes closer to the red cell unit as we increase the volume. In some of the parameters, we see a plateau. Some end up turning upwards as the higher the fill volume, the worse the quality actually gets. So we defined a really good bag size, an optimal bag size right there. And as you can see, the trend carries on. Most of those have the smallest delta, giving us the best idea of the quality. So I can tell you all about this at my poster, but in conclusion, we've determined that it is feasible to use mini bags, a small aliquot, for the quality control testing of red cells, which saves about $3 million, 9,000 red cell units per year. This needs to go through more testing, more optimization, but what we're hoping is that we found some very, very big savings in very, very small packages. Thanks.
My name is Namali Ratnamira, and this summer I had the opportunity of working with Dr. Agnes Lee at BGH on the long-term management of cancer-associated thrombosis using low molecular weight heparin. So cancer-associated thrombosis, or CAT, is when a cancer patient develops a blood clot. And according to guidelines, the standard treatment of CAT is 3 to 6 months of anticoagulation using low molecular weight heparin. However, there is actually very limited data regarding the dose, the type, and the intensity of anticoagulation, as well as the outcomes of patients. So at VGH, the current treatment of CAT includes continuation with full dose low molecular weight heparin, as well as dose reduction after 4 weeks of treatment. So our study looked at the outcomes of using low molecular weight heparin, including clot recurrence, bleeding, and death. So we conducted a retrospective chart review of patient records from 2015. And so this is just a summary of the results overall, but we found that among the 128 patients, only 13.3% actually had a clot recurrence, and there was actually a similar rate of clot recurrence among patients that had a dose reduction compared to those who did not. So overall, it didn't make too much of a difference. And we also interestingly found that patients who did have a clot recurrence actually were more likely to be diagnosed with certain types of cancer instead of others. Similarly, among the 11.7% of bleeding events that took place, there were actually similar rates of bleeding among patients who had a dose reduction compared to those who didn't. And so overall, you can see as well that there is 57.8% of the patients died, and the most common cause of death in patients was cancer, and only 1% was actually caused by VTE. And there is actually a large proportion of patients who are alive beyond 6 months after their initial diagnosis of CAT, and there was 72% of the patients were alive at 6 months, and then beyond 12 months after their initial diagnosis of CAT, 41.4% of the patients were actually alive. So if you're interested in finding out more details, please come check out my poster later. Thank you.
Good afternoon, my name is Anam from the Jeffries Lab. So in schools, hospitals, even shopping malls, a fast and effective first responder team or security team is needed in case of emergency. So just like in our body, in our cells to be specific, there are 3 pathways that constantly traffics and monitors the health status of our cell. So the MHC1 pathway monitoring the interior, the MHC2 pathway monitoring the cells surrounding, and a key player here is CD74 to keep that in mind because I'll come back to it. And the third one, an interesting phenomena called cross-presentation. So the cross-presentation is kind of like the merging of MHC1 and 2 pathways with the help of CD74. So here in a paper by Basha, the phenomena of cross-presentation and the significance of CD74 is being demonstrated within the cell here. So my job is to move forward and to analyze what happens out here on the cell surface. What are the exact peptide sequence that the T cells interact with? So by using a wild type and the CD74 knockout model, I tried to develop dendritic cells where the cross-presentation are found. And by developing them and by the end to send them to mass spec analysis, and I would look exactly at the sequence of the peptides. That being said, the bone marrow dendritic cell development is critical, and luckily that happened. So right here you can see that day 10 is when I have the most dendritic cells, and they're all grown up and for me to analyze and elute the peptides on the cell surface. And thousands and thousands of peptides have been found and sequenced, and I know exactly the sequence of the peptides. So now, what's the significance? How do I know they're so special? What's the sequence that makes them special to be cross-presented? And I'll only say that, so I am working towards, this is a novel and new vaccine strategy. So to know more, please come by my poster and I'll give you a complete detailed description. Thank you.
Good afternoon, my name is Guillermo Caballero. I'm a member of the Stranatica Lab, and today I'll be presenting the characterization of proteins involved in the peptidoglycan biosynthesis at the debutosome of Escherichia coli. So the main focus of the experiment was the study of PUP1B, which is a penicillin binding protein involved in the peptidoglycan biosynthesis. especially at the later stages of replication, and its interaction with various proteins such as CPOB, a negative regulator of transpeptidase, LNLPOB, a positive regulator of the transpeptidase activity, YCPB and LD transpeptidase, a PAP5, a carboxylic peptidase, and MOTG, a terminase, and various other proteins. So they are all involved in the synthesis. To do this, we had a protein method to extract and purify that will allow us to crystallize them. This was made by lysing and extracting the various proteins into various buffers that would have detergents that would solubilize the proteins and make them happy for concentration and purification by nickel column and size exclusion chromatography. Later on, we were able to crystallize by a sitting drop method, which involves putting proteins in specific plates at different conditions. This allowed us to crystallize and find the structure of, so far, 4 proteins, PP1B, the major structure, UB2H, the regulation domain found in PP1B, and LPOB, the positive regulator of the transmembrane domain, transeptase domain, as seen in blue on the PP1B structure, and PP5, a carboxylic peptidase. Further studies were made for the interactions were made by MST, which is microscale thermophoresis, which measures the binding affinity between PP1B and each of the interactors. This was made by measuring the fluorescence and how it would change between the concentrations through various having changed the concentrations. And you can see here that it shows a really high affinity by showing in the nanomolecular range. Thank you.
Good afternoon, everyone. My name is Helen, and I co-op the Carson Lab at the BC Genome Sciences Center. And today I'll be talking about the role of microRNAs in KDM5B and AML. So our lab primarily focuses on microRNAs. MicroRNAs are small, short, non-coding RNAs that help regulate gene expression, and their dysfunction can lead to cancers like AML. So like smart scientists that we are, we look for correlations. So using the Firehose dataset, we found that these microRNAs in purple, when highly expressed, actually correspond with low AML survival. Thankfully, all of these microRNAs are found within one cluster in the intron of CLCN5. CLCN5 is a chlorine channel, which is inversely correlated with KDM5B. And recent literature suggests that KDM5B, when highly expressed, negatively regulates leukemia cells. So our hypothesis is that these microRNAs from CLCN5 target the mRNA of KDM5B, silencing it, thereby allowing AML cells to proliferate. So what we did to prove this is that we looked at whether or not CLCN5 and KDM5B are indeed inversely correlated in AML cells using qPCR. Then we did a retroviral transduction of bone marrow cells with the microRNA clusters and a colony forming assay. And lastly, we used qPCR again to see if the microRNA is overexpressed and the KDM5B levels are suppressed. And bioinformatics was done throughout. So our results actually show that the bone marrow cells and AML cell lines behave in a way that we expected. KDM5B is present, however CLCN5 is not normally expressed in these cells, so we don't see them in there. The transduced cells are overexpressed in microRNAs, but instead of seeing a suppression in KDM5B, we actually see an increase, which is not what we expected at all. So our conclusion is that overexpression of microRNAs actually resulted in higher expression of KDM5B and also reduced colony growth. But whoever said that the path to true science ever ran smooth, not Shakespeare. Other potential targets we'll look into the future are ETV6 and MIB1, which are also inversely correlated to CLCN5. So as of right now, the microRNA key does not fit, but hopefully we'll find a door that fits in the future. Thank you.
Hi, my name is Hanju Brian Shim, and I'm a student at the Cote Lab over at UBC Hospital. And I just want to start by saying, we've been in school for a long time, and there's just one thing that's stuck with me over the years. The mitochondria is the powerhouse of the cell. Okay. But let's ignore the powerhouse, right? I want to look at the mitochondrial DNA. They do have DNA. It's small, closed circular, and multiple copies per cell. And this is what we call the mitochondrial DNA content. And that's what we've been looking at for the last 4 months. How do platelets impact whole blood mtDNA content? So in the whole blood, we have mononuclear cells shown on your right, and they have nuclear DNA and mitochondrial DNA. On your left, you have platelets, which have mitochondrial DNA, but no nuclear DNA. So when we define mitochondrial DNA content, we take the number of mitochondrial DNA copies and divide that by the number of nuclear DNA copies. You can see how this is confusing, as platelets would skew this ratio because they don't have nuclear DNA. And this is problematic because whole blood mtDNA content is associated with various diseases, aging, oxidative stress, and so on. So our objective was to identify the role of platelets in whole blood mtDNA measurements. So we took the whole blood from 44 volunteers, isolated for platelets and PBMCs, peripheral blood mononuclear cells, and we obtained platelet counts, leukocyte counts, the number of mitochondrial DNA copies, and nuclear DNA copies using the hematology system and multiplex qPCR. And what we found, surprisingly, we originally thought that platelet count was going to be a factor, but it didn't turn out to be. What actually mattered was the number of mitochondrial DNA copies per platelet, as well as the ratio of platelet to leukocytes. That was very interesting, so we decided to take it a step further. We simulated platelet to leukocyte ratio variation by spiking whole blood with purified platelets. And what we found was that you needed a threefold increase in the ratio in order to see any significant change in whole blood mtDNA content measurements. Now, what does this mean? Studies that are incorporating platelet to leukocyte ratio in their studying platelet to leukocyte ratios should be mindful of the range, so the min and max. If it's more than threefold, it can be a confounding factor. And also the average platelet mitochondrial DNA content we found in our sample of 44 was 3.7. I want to talk, there's a lot more in my poster. There's multivariable modeling, but all of that's actually kind of boring. So if you just want to come and talk to me about the clinical implications, and I really like volleyball, if you want to talk about that too, feel free to drop by. Thank you.
Hi everyone, my name is Catherine and I've been working at the Rossi Lab with Dr. Elena Gropa this summer studying skeletal muscle regeneration. In particular, I focused on paracrine cell signaling between two cellular populations involved in muscular regeneration, fibroadipogenic progenitors, or FAPs, and blood vessels. So, first let me explain what can go wrong in skeletal muscle regeneration. Simply, when you have a healthy muscle and injure it, we know that over time the muscle will heal and repair itself and typically go back to full function. However, for people with muscular dystrophy, we see that these damaged muscles don't actually repair properly. Rather, their healthy muscle fibers are replaced with fatty and fibrous tissues. And currently there are no cures for muscular dystrophy, only treatments and medications to slow its course. So, in order to be able to understand the mechanism of muscular dystrophy, it's crucial to understand what cell populations play key roles in muscle regeneration, namely muscle progenitors, inflammatory cells, fibroadipogenic progenitors, FAPs, and blood vessels. These four cell populations interact with each other intricately in order for healthy muscle regeneration to occur. And when this communication fails, we see degeneration happen. And so what we've been studying specifically is the crosstalk between FAPs and blood vessels and the necessity for it in adult muscle regeneration. Now, previous literature suggested that one specific protein, angiopoietin-1, was secreted by a cell population called paracytes. And angiopoietin-1 is known to play an important role in angiogenesis, the formation of new blood vessels. However, research by Dr. Gropa has found that FAPs and paracytes are actually two separate populations, and that angiopoietin-1 is actually secreted solely by FAPs, not paracytes. And there is evidence to suggest that it plays a role in muscle regeneration. So, To investigate the role of Ang1 in muscle regeneration, we took two types of mice, healthy wild-type mice, as well as mice which had their Ang1 gene knocked out, as well as and injected their tibialis anteriors with natexin, which causes acute damage. And then we collected the muscles at various time points post-injection to see how the muscles have regenerated or degenerated over time. And once we collected the muscles, we used various data analysis, primarily histology, to analyze this data. And what we actually found was that the depletion of Ang1 does indeed impair muscle regeneration, and that causes disorganized fibers in smaller diameters, which are characteristics of muscular dystrophy. And depleting Ang1 also has no effect on vascular density, but it does affect the vascular functionality of blood vessels. Great, so why do we care and why does it matter? So we know that angiopoietin-1 is critical for efficient muscle regeneration that affects vascular functionality, but we still have yet to understand how this happens. And importantly, Ang1 is an example of a cell population that isn't a muscle progenitor, but is still critical in muscle regeneration. And this illustrates the importance of identifying other targets or signaling pathways that can help us understand the orchestra of muscle regeneration and ultimately elucidate the mechanisms behind fibrosis. So if you'd like to hear more about this or see more results, then please feel free to stop by my poster later. Thank you.
Hi everyone, I'm Mahima from the Carson Lab, and today I'm going to be talking to you about myelodysplastic syndrome, or MDS. This has been mentioned to you briefly a little bit, but essentially it is a myeloid malignancy due to dysfunctional hematopoietic stem cells, or HSCs, which are cells that can differentiate into all other kind of blood cells. So just to give you a little background, much of the time HSCs must maintain quiescence, which is G0 in the cell cycle, and they only enter the cell cycle when mature blood cells are needed. And so disruption of this quiescent phase can propagate blood cancers or deplete HSCs over time. Additionally, our lab has previously found a microRNA called miR-146A, which is frequently lost in MDS, and our work in mouse models has shown that loss of MDS decreases HSC function. So my project aimed to improve our understanding of these findings. First, loss of miR-146A decreases quiescence and increases proliferation of HSCs, but can we detect these differences in quiescence and proliferation more easily using CD71, which is a surface marker known to stain cycling cells? Secondly, miR-146A represses this innate immune signaling pathway, and our RNA-seq data showed that TNF may be a potential driver of HSC dysfunction. So we were wondering whether loss of TNF or NF-kappa B based on this pathway can rescue the cell cycle defects seen in the miR-146 knockout. So in order to answer these two questions, we analyzed HSCs, first extracting the bone marrow from miR-146 knockout mice and isolating the HSCs using 4 cell surface markers and an additional marker called CD71 to stain the cycling cells. We then used single-cell FACS sorting to isolate the cells and measured the cell division timing manually, after which we cultured the cells for 10 days, monitoring the quiescence and proliferation. So we also did this for our 2 other genotypes. We did it for the TNF and miR-146 double knockouts to see if we can observe that rescue. So what we saw that we compared our CD71 expression to our initial manual measurements, and we saw that in the non-quiescent HSCs, there was a higher proportion of CD71 positive cells as opposed to the quiescent. So this suggests that CD71 is a good indicator of cell cycle quiescence. And then we wanted to compare it to our knockout and double knockout models. So we saw that in the knockout, there was a higher proportion of CD71 positive cells, consistent with the decreased HSC quiescence. And in our double knockouts, we see that there's a trend toward return to baseline, which suggests that there is a partial rescue of HSC quiescence. So that's pretty cool. And we hope that ongoing studies will be able to help us reinforce these results. Thank you for your time.
everyone, I'm Nicole from the Hancock Lab. For the summer, I've been working on studying the serving motility of Pseudomonas aeruginosa, specifically the adaptive antibiotic resistance mediated by the serving motility. I'll first start with a brief introduction on Pseudomonas aeruginosa. Pseudomonas aeruginosa is an opportunistic pathogen that's commonly associated with nosocomial infections, and it's highly prevalent in cystic fibrosis patients. And it has been found that on modified synthetic CF sputum media, which is the media mimics the lung condition of CF patients, that Pseudomonas exhibit an accelerated circular motility, which we term the serving motility. And what we are interested in is the antibiotic resistance mediated by this motility. To study that, we started with disk diffusion assays, and based on the result, we did liquid minimum inhibitory concentration tests and antibiotic incorporation assays. At the same time, we did literature search and RNA sequencing data matching. And based on the result, we did resistant gene screens, and we verified the result using qPCR. From the disk diffusion assays, we found that for all the listed antibiotics here, the antibiotics diffuse further on the control plates, which are represented by the blue bars, when compared to the serving plates, which are represented by the red bars. And similarly, for the antibiotic incorporation assays, we found that all serving plates, which are represented by the blue bars, show higher percentage coverage, meaning that they have more bacterial growth than the control plates. As for the liquid MIC, we found that bacteria grow in the liquid serving media don't necessarily show higher inhibitory concentration than the bacteria growing in the control media, which proves that the surface motility adaptation is necessary for the enhanced antibiotic resistance of the serving cells. And from literature search, we found 317 potential resistant genes, and among them 45 downregulated and 9 upregulated genes actually exist in the wild-type serving cells, according to the RNA sequencing data matching results. And we narrowed down our focus to 10 genes that will significantly alter the antibiotic resistance when knockout through the resistance screens. And we found 6 out of those 10 genes show the matching regulation pattern to the RNA sequencing data. In conclusion, serving cells are more resistant to antibiotics, and that may be due to the dysregulation of different resistant genes. Thank you for listening to my presentation.
Hi everyone, my name is Katina. I'm a summer student in the Algar lab as well as the Kazakadatu lab. I'm sure most of you are quite familiar with the traditional fluorescent proteins and dyes. Yes, they're extremely valuable in terms of bioimaging and bioanalysis. However, they do have their own limitations, namely they have limited brightness and they're quite susceptible to photobleaching. Because of these limitations, there's been a lot of interest in the development of new fluorescent materials such as polymer dots, P dots, and quantum dots, QDs, which are fluorescent nanoparticles with excellent optical properties that allow them to perform much brighter than the traditional fluorescent proteins and dyes. My project this summer is to employ surface modifications to first reduce the non-specific protein absorption of these nanoparticles, which is especially important if these nanoparticles are to be used in biological assays. Second, to improve their colloidal stability. That means when suspended in solution that they shouldn't be aggregating. And finally, to introduce new functional groups on their surface, which altogether will improve their biofunctionality. To do this, I first synthesized bare QDs and P dots. Then I synthesized compatible surface molecules that can then be attached to these nanoparticles. And finally, I evaluated the properties of these modified nanoparticles. Two types of surface modifications that I looked at, the first being dextran polysaccharide chains. These were used to coat the surface of P dots. Now, dextran has outstanding biocompatibility. They're highly water-soluble, and they've been shown to resist non-specific protein absorption. As well, they have free hydroxy groups, which allow for further downstream conjugation. On the QD side of my project, I was looking at hyperbranched polyglycerol polymers. They're non-toxic, highly water-soluble as well. They've also been shown to resist non-specific protein absorption much better than PEG, which is commonly used. And it also has multiple OH groups, which allow for the attachment of multiple ligands on the QD surface. So now some preliminary results. Here I've shown that in comparison with the bare P dots and the dextran-coated P dots, the absorbance spectra and the emission spectra are practically identical, which is a good indication that upon coating of these P dots with dextran, we're not changing the fluorescent properties of these nanoparticles. Also, using nanoparticle tracking analysis, which is a method that can be used to determine the effective hydrodynamic diameter of these nanoparticles, I've shown that with the dextran coating on the surface, you do see a slight increase in that diameter of these nanoparticles. However, this is to be expected, and this increase is not so large that we're worried about it affecting cellular uptake. Future directions that I would like to take with this project, if I had more time, would be to complete the synthesis of a dithiol-anchored HPG that can be used to cap the QDs. Next, to evaluate the efficacy of the dextran-coated PDOTs and HPG-capped QDs. That is to develop assays to test non-specific protein absorption between the coated and non-coated nanoparticles, as well as look at their improved colloidal stability. And finally, the functionalization of surface-modified nanoparticles for use in bioapplications. Thank you.
Hi, I'm Sam from the Hancock Lab, and for this summer I had a wonderful chance to purify potential antigenic proteins to combat one of the most costly diseases affecting the dairy and other agricultural industries in Canada today. More specifically, I've been purifying Mycobacterium avium subspecies paratuberculosis, aka MAP proteins, to prevent Johne's disease in infected cattle. So MAP is the sole infectious agent of Johne's disease in ruminants. It causes the decrease in body weight and milk production in infected cattle. The issues that are linked to this disease cost Canadian farmers $86.5 million every year, and of that amount, they cost Canadian dairy farmers $34.5 million every year. So the Revamp project aims to develop a vaccine that can save Canadian dairy farmers $31 million every year, and beef cattle farmers every year $52 million. So the Revamp project seeks to develop a subunit vaccine using reverse vaccinology, where they use the MAP genome to predict potential antigenic proteins and to purify them to be tested on cattle infection models. So my role in this project is to overexpress these MAP proteins in the heterologous expression host E. coli. Expression tests are conducted and analyzed using SDS page and Western blot probing for his tag, as you can see here. Proteins that overexpress are then purified using nickel affinity chromatography. Here, protein purification protocols are usually optimized in order to yield enough pure protein. The pure protein sample is then sent to Saskatchewan to be tested on cattle intestinal loop infection models. So far, I have worked on 18 proteins in total. 9 of them overexpressed, and only the purification of 3 of them yielded enough pure protein to be used for screening. I examined a few characteristics that can predict the expression level and protein purification yields of future vaccine candidates. So taking a closer look at the proteins, we noticed that membrane proteins and proteins with higher isoelectric points were difficult to overexpress. Those proteins, together with proteins with longer amino acid sequences, were also difficult to purify. So to know how I got to these conclusions, you can come visit my legendary, get it? Yeah, legendary poster later on, and thank you for your time.
Hi everyone, my name is Robert and I was a member of the Kim Lab this summer, and my project was looking at the link between periodontitis and thrombosis. So periodontitis is a gum disease and is an excellent model for chronic inflammation, and is extremely prevalent in North America, affecting half of the adult population. And it has been shown in previous literature to be related to thrombosis, which is the formation of blood clots and blood vessels. And here you can see an image of a set of healthy gums and an image of diseased gums here. So earlier this year, a master student in our lab, Vincenini, collected whole blood samples from patients with severe periodontitis and healthy controls. And he assessed them with an INTEM assay with a roto machine, where he found that there was a significant decrease in clot timing formation in the periodontitis patients compared to the health controls. So the mechanism that we think is causing these two diseases to be related to one another is that gram-negative bacteria from dental plaque, specifically P. gingivalis LPS, is binding to the toll-like receptors found on endothelial cells and inducing inflammation, but it's also found on platelets as well, and we think it is affecting the coagulation with the platelets. So he wants to show an in vitro experiment with endothelial cells where we expose them to P. gingivalis LPS, where we expected to see higher amounts of active MMP2 and MMP9, and I'll get into that in just a bit, because we went back to the whole blood samples and isolated the plasma from these samples and tested them for circulating coagulant factors, specifically factor 13 and fibrinogen, where we found for both cases significantly higher levels of these factors in the periodontist patients compared to the controls. So for the endothelial cells, they were plated in 6 walled plates and starved for 24 hours before exposing them to different concentrations of LPS, and then we collected the cell supernames and assessed them for MMP activity in gel zymography, where we didn't see any significant results, as you can see here. So we concentrated the media by a factor of 10 and plated them in a quench fluorescent 24 assay. As you can see here, the concentrated show a lot of MMP activity, while the regular media sample showed very little, almost half. So future directions for the project is to repeat the zymograms with the concentrated cell media to see the MMP activity being shown in the assay, and to characterize TLR4 signaling and endothelial cells by Western blotting. Thank you.
Good afternoon, my name is Jillian Speakman. I work under Dr. Tom Neville at Vancouver General Hospital, and my project is a retrospective chart and database review about GPI deficiencies in hematologic diseases. Paroxysmal nocturnal hemoglobinuria is an ultra-rare disease where the blood cells are deficient in GPI-linked proteins, and this can lead to hypersensitivity of complement-mediated destruction. And PNH is often suspected when patients present with abnormally low blood counts, elevated LDH, or unexplained thromboses. So all of the 55 patients within this cohort have a PNH clone to some degree, and they were further categorized into 4 separate groups based on their initial diagnosis. These 4 groups include aplastic anemia and PNH overlap, classic PNH, and MDS, and the median clone size for the 4 groups together was 4.5%. Some of the treatments administered include immunosuppressive therapy, stem cell transplantation, and a drug called eculizumab, and it's important to know that 11 of these 55 patients did receive a stem cell transplant, and 10 of those 11 are still alive and well with a median follow-up of 27 months. And this is a graph showing the hemoglobin results for all of the 4 groups at diagnosis and at 1-year follow-up. And an interesting point is that even though the classic PNH group has the highest median hemoglobin at diagnosis, none of these patients did have a normal hemoglobin at presentation. And also this decline here that you see at the 1 year for the classic PNH group is attributed to progressive marrow failure, while the improvement in the counts at 1 year for the other groups is because of the effect of the immunosuppressive therapy. This graph is showing the platelet results at diagnosis, and in this classic PNH group, 50% of the patients presented with normal platelets, but this did tend to decline at the 1 year follow-up. And while I don't have time to show all of the data that was collected, the white cells were similarly normal at presentation and tended to decline after 1 year, but in the other groups, the platelets and the white cells did improve over time. This graph is showing the normal versus elevated LDH results at diagnosis, and as you can see, the overlap group and the classic PNH group had almost all of their patients with elevated LDH, meaning that they were experiencing a great deal of hemolysis.
And as for the MDS group, this 40% of patients with elevated LDH can be attributed to tumor burden. And some conclusions that can be drawn based on the data that I've shown above include that at diagnosis, classic PNH patients were always anemic and had elevated LDH levels, the latter being distinctly uncommon in the aplastic anemia group. Platelet counts were frequently normal at diagnosis in the classic PNH group, but tended to decrease over time, consistent with evolving marrow failure. And none of the other 3 groups patients had normal platelets at diagnosis, and all of their counts did tend to improve over time. As for treatments, immunosuppressive therapy was used effectively in all of the groups except for the classic PNH group. Eculizumab was only used in the classic PNH and the overlap patients, and stem cell transplants were highly successful and were used in all of the 4 patient groups. Thank you very much.
So what do you think? We give them all a round of applause for great. So I'm only disappointed that I didn't get to do that. So I'd ask all the scorers to complete these, all the students complete the scoring, please, and you can hang on to them or pile them that way, as you wish. Okay, so we're going to move on to the next part of the program. That's not there. So I'm going to ask actually Dieter Brumman just to describe or talk about Neil McKenzie.
So hello. So it will be my honor to, to honor this year's recipient of the Neil McKenzie Mentorship Excellence Award. So who was Neil McKenzie? Neil McKenzie was a postdoc in my lab. He arrived in May, I think, 2013. He came from Scotland, Edinburgh, and he was a really Scotsman. And he had definitely 2 loves. I mean, that was science and outdoors. And the outdoors were maybe even a little bit more than the science. So he spent pretty much every weekend in the mountains or anywhere in the outdoors, on the beach, but mostly I think in the mountains. So he was an excellent scientist, a postdoc in the lab. So he worked on a topic like the mineralization of vascular tissues, and he made quite a lot of progress in this project. But unfortunately, he had a fatal accident on January 11th in 2015, just 1.5 years in the lab, and he died together with 2 of his friends. So it was a big loss for all of us at UBC, and particularly also in my lab. And we generated essentially this Neil McKenzie Mentorship Award, because Neil was also heavily involved in mentoring students like you, summer students. And of course he mentored a lot of outdoor people, learning how to climb mountains, how to hang on the ropes. And it's really a big loss for us. And I think we keep his memory with this Mentorship Award alive. And I think his parents are also here. Is it? Yeah. And so you are very welcome again to see you. And I think we all miss Neil, but we keep him alive in our memory. So, and this year's award is handed over by you, I guess.
Right.
Who is it?
So these are the nominees. There are many great mentors, obviously, here. These are the ones who were nominated by colleagues and students and technicians and anybody else who wanted to, you can see. And this is just some of the, I just wanted to read just a few of these comments that were made in the nomination forms, that the mentor was available, approachable, attentive, never doubts my abilities, respects my perspectives, phenomenal listener, no matter how busy, never fails to find a reason to compliment, offers constructive criticism, promotes a positive and engaging environment, nurturing, caring, made me feel at home, encouraging, empowering, non-judgmental, reliable, never blames, approachable and responsive, eager to teach, generous, open-minded, motivates, inspires, patient and dedicated, a clear communicator, supportive, builds up my confidence. And the one I like the best is pushes me to step out of the comfort zone, but always a half step behind to provide support. So these are the kinds of people we're talking about here, and really represents what Neil was all about. So the winner of the award this year, and it was obviously very difficult, is Ume Amara. Ume? Do you want to say anything?
Yes.
Please, go ahead. First, why don't you go ahead and we'll give you the award after.
Thank you. I would like to first thank God for gracing my life with an opportunities which wasn't in my hand. I also express my thanks to CBR. CBR is a place of scientists with a great potential, and I'm privileged to be part of the CBR. I would also like to thank my mentor, and his mentorship reflects in my success. So thank you, Dr. Kim. I can't see you. So I would also like to thank the summer student who nominated me, who gave me an opportunity to know them, who gave me an opportunity to impact their work. During this mentorship experience, I tried to reflect their strength, the strength they didn't see in themselves. So on the other hand, I tried to be very honest. I tried to give them feedback by not creating a major scars. So mentorship, the magic of mentorship is the people we meet and what we get from them in return. So thank you, CBR. Thank you, my lab. Thank you, summer students. And thank you for giving me this award.
Right here. 3, 2, 1. I'll do one more. 3, 2, 1. Thank you.
Okay, so hi Jamie, nice to meet you. So our keynote speaker, for those who were a little late, the keynote has changed. And I won't go through that little story, but we're really actually honored and pleased to have Jamie Matthews here. Now, I don't know Jamie, but he sent me a bio that's a great bio. And the boring stuff that he was born in Chatham, and I know where Chatham is, which is pretty crazy. I used to go there as a kid.
There's 2 Chathams. There's Chatham, Ontario.
Ontario.
Chatham, New Brunswick.
That's the one I know, Ontario. Yeah, the Chatham, New Brunswick is less well-known probably. He completed his BSc at the University of Toronto, a master's and PhD at the University of Western Ontario, or Western University as it's now known. And then he held various awards, did postdoc fellowships at UBC, and then was at University of Montreal before joining faculty at UBC in 1992, where he is now a professor of astronomy and astrophysics. And he's really had an incredible journey, and his bio is like amazing. But let me I'm going to just read a couple of things that he wrote. Okay, so he describes himself as, quote, an astrophysical gossip columnist who unveils hidden lifestyles of stars by eavesdropping on the music of the spheres. Now, I have absolutely no idea what he's talking about. Okay, but my understanding is that he sort of leads missions with NASA, NASA scientists, and he searches for Earth-sized sort of exoplanets with, as he says, habitable zones of their stars that might be homes for aliens. Now, one of my favorite movies is Contact. Okay, I love that movie. All right, so I can really relate. So for his many achievements and his leadership in these fields, he's won a whole wild list of awards, including he's a member of the Order of Canada. Now, that's not all, though. Really, he's dedicated to communicating through education and teaching and outreach, and he does work in the downtown east side. He's all over the media on CTV, CBC, CNN when they're not covering Trump stories. You know, he's really out there. And he's a writer and he's a producer and really a celebrity. And many of you will recognize Jamie from his media adventures there, where he might be caught sporting various strange and intergalactic costumes like the one I'm showing here. So before he takes the stage, because it's at the bottom of his bio, he says, I can't help mention that he wrote 30 years ago in Discover magazine, quote, exploding star contains atoms from Elvis Presley's brain. Scientists confirm that the king of rock and roll lived in another galaxy 170,000 years ago. So with that, Jamie. Okay.
Somebody knew I was coming, obviously. Rubber chicken, classic. Allow me a few minutes to set up.
Does anybody want to stand and stretch? Now's the time.
And maybe if we can have this bank of lights off, because I have astronomers hate light pollution, and I'll be showing some dark slides that might be better appreciated. You don't need to see me.
Does somebody know use those lights right behind you? I'm not sure what number, Ross. Try one of the numbers, 3 or 1 or...
Oh. And I know the set, you know where the power bar is?
Yeah, it's right here.
Thanks. Actually, I could probably keep the lights over here. There's a bunch of presets. Okay, there's a bunch of presets. That's definitely not the one.
Hi, I'm IPA guy. Just want to know, are you a walker or are you just...
No, I'm going to stay right here.
Is that too dark?
If we can bring up some lights for the crowd. Yeah, so we're just... Normally I have... that's pretty good. Yeah, I'm happy with that. We got to plug into... I have a...
Mine, that's mine.
Oh,
put that away.
And I have audio.
We have audio, by the way.
I should point out, you are Ed, by the way, aren't you? Because we've never actually met face to face. So I should point out that one of Ed's favorite movies, Contact, that basically Jodie Foster, I think, is the best performance ever portrayal of an astronomer and a scientist in a popular Hollywood film, and I would say that stands today. And we just celebrated the anniversary of the debut of the movie Contact recently. And the Elvis story, I didn't write that in Discover. I was quoted by journalists who were on the mountain when I was in Chile when the light from the brightest supernova in 400 years arrived on Earth. And I was explaining to them that my work was maybe not the stuff of which headlines would be made, but if I wanted to make headlines, I would submit anonymous tips to the tabloids, and I gave them a few of them, like desperate astronomers' fake supernova explosion in desperate attempt to attract girlfriends. And one of them was I was trying to explain the fact that almost every element in your bodies and in what we're wearing and even in this planet were generated in the cores of stars, and most of that was distributed into space to form future generations of stars, including ours, in supernova explosions. And so, as Carl Sagan said, we are made of star stuff. And I was trying to get that idea across, and I was reversing it by saying so Elvis Presley's brain contained atoms from a supernova, so I said, the headline I'd make is a stellar explosion contains atoms from Elvis Presley's brain. And that explosion was taking place in a satellite galaxy of our own, 170,000 light years away, so hence the subtitle, Scientists Confirm the King of Rock and Roll Lived in Another Galaxy 170,000 years ago, because it takes that long for the light to reach us. And it's maybe appropriate because it's just the anniversary of the death of Elvis Presley. And I can remember where I was as a a student and making money. I grew up in a poor family in Chatham, Ontario, and so I worked on a truck assembly line in an international harvester plant in Chatham in the summers to earn pay for tuition and expenses to be at the University of Toronto. And I was on the assembly line when the news came out over the radio, and so I can kind of remember that. Now, I am a very poor substitute for your scheduled speaker, Farah, and I apologize for that, but I have incorporated into my talk an introduction to, an introduction to an inspiring Canadian woman who's a student here at UBC. Now, Ed had mentioned that I'm involved, a writer and a producer, truncated of lots of things for media, and one project that I've been involved in is called Space Suite, and it runs on British Columbia's educational television network called Knowledge, and we've had Space Suite 1 running for a couple of years, and Space Suite 2, and Space Suite 3 will debut this Sunday night, if you want to watch on Knowledge or go to their website. And I just thought, as a test for the audio and the video, and as a demonstration of this, I'll share with you one of our Space Suites. They're like space astronomy music videos meant to run between shows. And here's one that I think you'll enjoy, if I can get it started. Yeah. I mean, it was meant to start automatically, so let's see what happens here. Yeah, there we go. I should tell you, to relate to the topic of this conference, there's a lot of blood in the Milky Way galaxy, at least here, and hopefully elsewhere. And this is a real supercomputer simulation of what we expect to happen in a few billion years. So that's an example of my attempts to bring rocket science to the general public. So, and again, I apologize, not only am I poor substitute for Farah, but I am a fish out of water when it comes to your research topic. I'm a rocket scientist, but watching the last of the student presentations, I couldn't get here any sooner for all of them. It blows my mind, so it's rocket science to me. Today I'm going to tell you about the search for planets that might be able to support life, that satisfy at least a few of the conditions for life as we know it. And I'm going to take you into space in a sense, and there are astronauts who travel into space. Many of you will be familiar with retired astronaut Chris Hadfield. I'm an astronaut, but I'm a virtual astronaut. I explore other worlds without ever leaving the surface of this world, although I do use satellites and space observatories which are at points far beyond the greatest reach of any human in our history. But before I take you on a virtual voyage through space, I'm going to take you on a trip back in time. So we're going to charge up the flux capacitor with plutonium on our DeLorean, and hit 88 miles per hour, whatever that is in kilometers per hour, I've forgotten the conversion, and we're going to go back in time. Now, in the original movie Back to the Future, their first trip was back to November 5th, 1955. I'm going to take you much further back. We're going back to January 7th, 1610. We're going back to the early 17th century. And here's a genuine painting, painted in the 17th century, depicting perhaps the medieval stargazers with the rising moon on a clear night. Now, on the night of January 7th, 1610, there were only 6 planets known to all of humanity. The 5 that you could see without a telescope looking up: Mercury, Venus, Mars, Jupiter, and Saturn. And this is really a family portrait of those naked-eye planets taken by an Ontario astrophotographer, amateur astronomer. And then the one planet that you can see without a telescope by looking down, the one we're on, the Earth. Now, on the night of January 7th, 1610, no new planets were discovered. The reason I picked this date in history is because this was the night that Galileo Galilei pointed his small telescope, state of the art at the time, although primitive by today's standards, at what was called a wandering star, called Jupiter. And wandering stars were things that looked like stars, but they didn't stay fixed in the patterns of the constellations. That's where we get the word planet from the Greek word for wanderer. And Galileo, pointing a telescope at this one wandering star, found that it wasn't just a pinpoint of light in the sky. It was a world. And it had even tinier worlds, looking like pinpoints of light, whirling around it in orbit, the 4 largest moons, which we refer to as the Galilean moons. And there on the right, you see the sketches that he made of what he saw through his telescope from night to night, from hour to hour, as these moons moved in their orbit. Now, they all appear to be moving kind of side to side, like beads on a string or on an abacus, but that's only because we're looking at the orbit sideways. They're almost circular orbits, but the Jupiter orbits in the same plane around the sun as the Earth does, roughly, and its moons orbit in roughly the same plane. So we have a sideways view of everything, and we have no depth perception, so it doesn't look like things are moving closer or further away. It just looks like they're moving side to side. So this was the beginning of the telescopic era. And in the next 4 centuries, telescopes got bigger, and the things that we put at the focus of a telescope got more sensitive. We went from the eye to photographic emulsions to digital detectors. So after, say, 377 years, how many planets did we add to the inventory? 3. 3! After almost 400 years. And to be honest, we actually only added 2, because we nasty astronomers downsized Pluto to the status of a dwarf planet in 2006. And I should point out, by the way, dwarf planet isn't a loophole to keep Pluto in the planetary club. You got to say the 2 words dwarf planet as a single phrase. It's like hot dog. A hot dog is not a canine that needs air conditioning. A hot dog is a hot dog. Dwarf planet is a dwarf planet, not a planet. Where do we stand as of this afternoon? And I've been doing everything at the last minute because I'm doing this on like 4 hours notice. But this is one thing that I would check at the last minute, because the tally is changing day by day, week by week. You have to check to keep up. As of this afternoon, as of about 2 hours ago, the total number of planets known to humanity, 3,653, and almost 5,000 candidates, of which I would estimate 90 to 95% of those will stand the test of time. There's 5 or 10% that are what we call false positives, signals that are mimicking a planet. We just don't know which 5%, and so we lump them all in the standard of category of candidates. But it's no exaggeration to say that as of this afternoon, we know of almost 8,000 planets in the universe. Now, that's a small number compared to the several hundred billion stars that are in our Milky Way galaxy. And as you saw in that space astronomy music video I showed, almost 1 trillion galaxies in the observable universe. But compared to a handful that we knew only 20 years ago, this is an explosive increase. And in fact, planetary science has for the first time become a statistical science. We are actually doing our first census of the galaxy to understand the nature of planets and planetary systems, and that is teaching us about our own home planet, the Earth, and our planetary system in an objective way for the first time. It's a revolutionary time. And maybe it's appropriate that I started out with in the 1600s because the reason that I can use revolution in the sense that I just used it, that word, is because Copernicus, Nikolai Copernicus, the Polish astronomer, who had promoted the idea that the sun was at the center of the solar system. He wasn't the first to suggest this, but he put the arguments together in very compelling ways and at a time when other pieces of the puzzle in terms of observations were falling into place. And so his book, published posthumously by his assistant Rheticus after he had died, and by the way, here at UBC, one of my colleagues, Dennis Danielson, is one of the world experts on Copernicus and has a great book that you can pick up at the UBC bookstore called The Last Copernican. I can highly recommend the human story of these discoveries. But the title of the book in Latin was De Revolutionibus des Orbis, of the orbits of the planets. And before that, the term revolution just meant circular or orbital motion. But because of the impact of Copernicus' book, people started using the word revolution in the sense of an overturning of scientific and political and social and theological thought. So it's embedded in our very language. And who knows what will be in the language decades or centuries from now based on the revolutionary discoveries that are being made right now, not only in my field, but in your field by you. Now, in the days when there were only a handful of planets that were known and a small number of moons and there were no computers or calculators, people kept track of the motions of the known planets around the sun and the moons known to orbit those planets with a clockwork mechanism called an orrery, named after some long dead British dude, actually named after his title, since his name was Charles Boyle, and we don't call them Boyles, thank God. We call them orreries. He was the Earl of Orrery. And these are real orreries, originally cranked by hand and eventually motorized. That won't cut it today. Let me share with you a modern digital orrery. Now, what you're seeing here, all those sets of circles are orbits. Think of them like dartboards. And at the bullseye of each of those dartboards, there is a star. But you can't see the star because if I included the stars in this animation, it would get too cluttered. Moving around on the circles on each orbital dartboard are planets. And the planets have been expanded in size compared to their orbits, otherwise you wouldn't see them, but their relative sizes to each other are shown. The only other cheat is time has been sped up. Days are flashing by in seconds. And it's almost hypnotizing, which is why I've got Flight of the Bumblebee playing, so you don't fall asleep. It's late in the day, and I might not be keeping awake. And as we zoom in on some of these systems, it's almost looking like looking at an animation of electrons whizzing around invisible nuclei. And this is just the tip of the iceberg. This is just a subset of the planets and planetary systems that we know today. Now here's a police lineup of a few planets, including 3 of the usual suspects. And in this picture, I ask you to ignore the colors. Don't ignore the colors. The artist put them in there so that you pay attention to this, but the colors convey no information. It's only the relative sizes of these spheres. And at the top row, on the left is the largest planet in our solar system, Jupiter, gas giant, almost a dozen times the diameter of the Earth, 320 times its mass. On the third row, second from the right, Neptune, an ice giant, about 4 times the diameter of the Earth, about 10 times its mass. And then at the bottom left is the planet we know and love, the Earth. And you can see that even in this small subset of planets, that we were approaching planets the size of the Earth. And as I'll show you, we've gotten down to Earth-sized planets and even smaller than Earth-sized planets. Now I have to apologize in the time that I'm taking, and I'm probably going to take more time than I should, and I know there's a barbecue right after me, and it's, I think W.C. Fields was once claimed to have said never follow children or dogs on stage, and I would say never speak before lunch or a barbecue. But I have places where I can stop, and I'll look to Ed, and he can yank me off stage if necessary. But in the time that I do have, I'm not really going to be able to tell you much about how we have discovered planets around other stars, but I can tell you that the search started right here at UBC and UVic in the late 70s and early 80s by two Canadian astronomers, Gordon Walker and Bruce Campbell, not the Bruce Campbell of Evil Dead Army of Darkness fame, for those of you that are movie fans, another Bruce Campbell. And the very first planet outside the solar system ever to be discovered was actually discovered here and announced in 1988, but nobody believed the result at the time. And the two astronomers were Canadians, so they're polite, they didn't insist. And so you don't read about that in the textbooks, but everybody in the planet hunting community knows the story. And if there is ever a Nobel Prize awarded for exoplanetary discovery, Gordon and Bruce would be in the running to be considered to share in that prize. So I'm not going to go into those details, although I will take a little detour and you'll get a brief explanation of one of the techniques that we use. But I want to cut to the chase, because there are lots of reasons to hunt for planets, to understand the nature of our planet, but one exciting reason is to look to see if there's life out there. And the question of whether life exists beyond Earth, for some people, it's basically just science fiction. And for others, it's a fairy tale. And to be honest, it is actually kind of a fairy tale. The search, at least, for habitable planets has elements of Goldilocks and the Three Bears. Except my version is called Jamie and the Three Planets. And to tell you this story, I've got to come back closer to home. But before we come back to Earth, I'm going to stop at one of our neighboring planets, Venus. Venus, by astronomers, is dubbed the Earth's sister world because it's almost the same mass and size as the Earth, and it has a thick atmosphere. And for a long time, it was believed that beneath the clouds which shrouded the surface of Venus from view, and still do to visible light, that there would be life. That was where scientists were betting on life in our solar system, much more than Mars, which had better PR due to Percival Lowell's mistaken belief in canals on Mars and his ability to tell the story very eloquently. But unfortunately, although Venus has a relatively romantic name, we now know it's a hellish place. Beneath those clouds are raging volcanoes, sulfuric acid rain. We're worried about acid rain in our environment. They have sulfuric acid rain. Yeah, it evaporates before it hits the surface, but the reason it evaporates before it hits the surface temperature is 500 degrees Celsius. That's higher than the melting point of lead. If you were to bring a lead vase to Venus, I don't know why you would do that, but if you did, it would melt. And so for liquid water oceans and for life of any kind that we can conceive, Venus is too hot. What about Mars? Now, when people think aliens, often it's synonymous with Martians. And Mars does have things that sound conducive to life and similar to what we have on Earth. It does have an atmosphere. It does have water, but the atmosphere is very thin. The air pressure on Mars at its surface is about 0.6% of what we're breathing now. It's mostly carbon dioxide. The water is almost entirely frozen in polar caps and in kind of a permafrost in the Martian rock. And the average temperature, 60 degrees below zero. And on a balmy day in the tropics, the temperature can shoot up to like minus 20. And there are times when it can actually be above zero, but very rarely. And so for microbes, for hardy creatures that we would refer to as extremophiles, the kinds of things that eke out an existence in Antarctica or in salt lakes in the Atacama Desert of Chile and so on, then maybe there are microbes and bacteria, and that's the kind of thing that we're looking for evidence either surviving today or having existed in the past. But for broad liquid water and complex life, and when I say complex life, a mushroom is complex life, that Mars is too cold. And then nestled between Venus and Mars, their orbits, is the Earth. and with an average temperature of about 15, 16 degrees Celsius, below room temperature. And of course, how that temperature is changing with time is a great issue of concern at the moment. But in terms of liquid water oceans and life, the Earth is just right. We're here. The Earth is in the zone. It's in what we call the habitable zone. That's the range of distances from a star, in this case the sun, where the temperature at the surface of a solid planet would allow liquid water oceans and possibly life. So the temperatures between the boiling point and the freezing point of water. We nickname that the Goldilocks zone, because if the planetary bowl of porridge is too close to its star, it's too hot. If it's too far away, it's too cold. And if it's in the zone, it's just right. Now, it's also kind of like being around a stellar version of a campfire. If you're too close to the flames, you burn yourself. If you're too far away on a frigid night without protection, you'll freeze to death. But if you're at the right distance, as long as that fire is maintained, you can survive the night. Now, not all stars in our galaxy are campfires, like the sun. Some are much hotter. They're more like raging bonfires. But the majority, 3/4 of the stars in our Milky Way galaxy, are these dim, small, red stars that we call M dwarfs or red dwarfs. It's like kindling, or like a little match, and you have to huddle close to that match to stay warm. And so the habitable zones, the Goldilocks zones, are hugging close to those stars and are fairly narrow. Now, my colleague Ravi Koparapu, it's at Penn State and the NASA Goddard Space Flight Lab, and I are trying to mount a campaign in our community to change the name to something a little more representative. Because habitable zone could convince non-experts that any planet in that zone could support life, and that's not the case. Really, it's just any planet in that zone potentially could have liquid water on its surface. And that is one of the things that we would look for life to evolve. And this is just a plot showing the star's mass relative to the sun. So 1 is the sun's mass, and distance on a log scale in Earth-Sun distance is the technical term for that is the astronomical unit. And that rainbow pattern you see, a kind of an echo of the Pride weekend that we had recently here in Vancouver, are basically different definitions of the boundaries of the habitable zone, and compared with the orbits, the distances of the planets in our own solar system from the sun. Now, so although the habitable zone doesn't mean guaranteed to support life, finding a place that could have liquid water on its surface is a start. But only a start, because there are many factors in the habitability of a planet. That depends not only on the planet and its environment, but its host star, the other planets in its system, and how they interact. And this are showing just a few of the interconnected factors, but even this is just the tip of the iceberg. Here's a more realistic thing, and this isn't something that just made up at random. Each one of these squares and ovals and triangles represents a parameter which has to be included if you're going to try to evaluate the habitability of a world. And even this is probably insufficient to encompass all of the factors. But let me break it down to something simpler. Here's a Venn diagram. In mathematics and physics, we like Venn diagrams that show where they overlap, they show the commonality of things. And here are basically kind of the 4 big picture requirements for life. At the upper left, you got to have the right ingredients, the right raw materials, and people in astrobiology refer to these as schnapps. Probably people who spend a lot of time in Europe and like to tipple and schnapps and Jägermeister and so on. But the schnapps in this case is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The 6 elements which are key to life on this planet, and the star of it, and the one why it's at the beginning of the list, is carbon. And then at the upper right, energy. You need some source of energy, whether it's sun or starlight for things like photosynthesis, whether it's geothermal energy, tidal energy, but electricity through lightning strikes, some form of energy. At the lower right, the right physical and chemical conditions and the right atmospheric pressures and things like that. And then finally at the lower left, a solvent and a transport medium. And by far the best solvent and transport medium for biochemistry is water, just as by far the best building block for molecules for life biology is carbon. Now, I'm going to mention Star Trek shortly, and any time that I talk about this, my students, if I talk about carbon and water being the best building block and the best solvent and transport medium, somebody will put up their hand and say, what about silicon? And what about ammonia? And the silicon always comes, people tend to know about it because there was a classic Star Trek episode where they discovered silicon-based life that kind of looked like moving rocks, called the Horta. And I'm sorry that I actually know that, but... But I should point out that yes, silicon is second best in terms of its biochemical properties, and ammonia is second best. But second best in the same way as that if I were in a one-on-one 100-meter dash against Usain Bolt, I would come in second. And I would come in a distant second. Now, to be honest, ammonia is a little better than I would be against Usain Bolt in a race, but still would be left far behind. And then we do live in a big universe with lots of stars, and I will argue evidence points to many, many planets. So maybe there's some place where the rolls of the die have led to silicon-based life and ammonia oceans. But if I had to bet on it, the majority of worlds that host life, I would expect essentially organic life and water oceans. So here is a tally of some of the small rocky planets that are roughly Earth size that we knew of. As of 2 years ago, the number was a baker's dozen, and we had just added to the list the first Earth-sized planet in the habitable zone of a sun-like star. That's what the G, K, and M are temperature classifications astronomers use, and a G-type star is a sun-like star. And so other than the Earth, this was the first one added to that list. And then about a year ago, we found an Earth mass star around our nearest neighboring, an Earth mass planet, I apologize, around our nearest neighboring star, Proxima Centauri, and the planet's called Proxima b. And I was involved in that discovery and characterization. And then as of about 4 months ago, there were 7 Earth-sized planets around an ultra-cool M-dwarf star called TRAPPIST-1a, and 3 of those are formally in the habitable zone. So there's a few more that have been added, and I'm sorry I didn't have a chance to incorporate them on 4 hours' notice, but I can talk to you about those afterwards over the barbecue as long as I can stick around. Let me just come back to our nearest neighboring star, and the moment what would be our nearest neighboring Earth-sized planet, Proxima b, which potentially could have life, and it's encouraging to know that of all the stars in our galaxy, that there is a planet that might harbor life around our nearest neighbor. I don't think we were ever so optimistic that might happen. I don't think even Gene Roddenberry in Star Trek was that optimistic. Here's a picture of Proxima Centauri. To point out, it's not that bright star, just the lower center, that's Alpha Centauri. It's not the bright one that's off to the left, that's Beta Centauri. It's right at the center, surrounded by a little red circle. So that's where it is, and there's an artist's conception showing what it might look like from being in orbit around planet Proxima b with the red dwarf star Proxima Centauri a in the background. Now, I'm not going to talk about how we discover these planets in detail, but I can tell you how we don't. Because most people might think, given the big telescopes we have and the sensitive detectors and the instruments we have, that we could just take a picture of an Earth-sized planet around another star. And Proxima Centauri is our nearest neighbor. It is a red dwarf star. It is a member of the largest demographic in the population of our stellar City of Stars, Metro Milky Way. And I was calling it City of Stars long before Ryan Gosling was singing it in La La Land, I should point out. So I should charge them for some licensing fees. So this would be the best chance. It's our nearest neighboring star beyond the sun. But taking a picture of an Earth-like planet around Proxima Centauri, which is 4.2 light years away, so it takes light more than 4 years to get to us from this system, is like taking a picture of a grain of sand sitting on a person's finger. And there's the grain of sand. It's about 0.3 millimeters long, except the person and the grain of sand are not standing right in front of the camera. The person with the grain of sand are 1,000 kilometers away. And I can't even show it as a pinpoint of light because the person and the sand don't reflect enough light to be seen at that distance, so I just put the arrow in to let you know where they were. So can we photograph an alien Earth around any nearby star? Not yet, but we're working on it. And even in the infrared part of the spectrum, where stars are often dimmer and the planets are glowing due to their own warmth, we have a better contrast effect, and that's probably where we will get the first pictures of an Earth-like planet around a Sun-like star, but we're not there yet. Now, I want to share with you, I said that I was a poor substitute for Farah, but that I would introduce you to another inspiring woman, and here she is, Michelle Kunamoto. Michelle graduated a little more than a year ago from UBC with her Bachelor of Science in Honors Physics and Astronomy. And before she had graduated, before she had any letters after her name, she had discovered 4 new planets outside the solar system in data from the NASA Kepler satellite, which were not found by NASA experts. Now, Michelle is a Star Trek fan, and so that's why I have Going Boldly there, but she didn't go boldly where no one has gone before. She went boldly where where others had gone before and found what NASA experts had missed. And here's the police lineup of the planets that she found, along with 3 usual suspects from our solar system, Mercury, the Earth, and Neptune. And you would think the excitement would be about the Earth-sized planets. That usually is the case, but not in this case. The exciting planet of these 4 is the Neptune-sized one. And here's why. Let me show you another orrery, a digital orrery, but this time now with only 5 planetary systems, including the Earth orbiting the Sun. That gives you the clock, by the way. Once the Earth has completed one circuit around the Sun, a year will have elapsed, so you can see how time is advancing. And of these 4 planets, it's the ice giant size, the Neptune-sized planet, KOI 408, which is actually in the Goldilocks zone of its star. The KOIs, by the way, stand for Kepler Object of Interest, this one is inside its star's habitable zone. Now, even a warm Neptune, that's what we've dubbed this planet, it likely wouldn't have oceans. It wouldn't have the right kind of structure to have a solid surface on which you could have liquid water oceans. But remember I talked about Galileo seeing these large moons orbiting around Jupiter with his telescope? All the giant planets in our solar system have large moons, and here are the 4 Galilean moons shown to scale alongside Jupiter, which is so big compared to them that you can't fit all of it into the slide. So I've just put a little bit in, and I've happened to put the part that has this great red spot on Jupiter, which astronomers have cleverly named the Great Red Spot on Jupiter, which is a hurricane, or a cyclone, an anticyclone, which would swallow up at least 1 or 2 Earths. Now, so giant moons, large moons are almost certainly a natural byproduct of the formation of giant planets. Now, you might remember this place, Pandora, from this little movie called Avatar. Jim Cameron is working on Avatars 2, 3, and 4 right as we speak, and I think the first one isn't due out until 2019 or something. But Pandora is not a planet. You know, this place with the big blue creatures with long tails and unruly hair, it's not a planet. It's an Earth-sized moon of a giant planet. And here you can see from a scene of the movie, it's the giant planet around which it's orbiting. And so Michelle has essentially found a place where we could look for a real-life version of Pandora. Now, to give you a little backstory and to give my voice a break, I'm going to share with you a global news report that came out on her graduation day describing the research and maybe filling in a few of the blanks that I haven't had a chance to tell you about.
Start 1, 0, and liftoff of the Delta II rocket with Kepler. For 4 years, the Kepler Space Telescope had been fixated on one sector of the cosmos. It stared unblinkingly at the Cygnus constellation, trying to detect the slightest variation in star luminance. An undergraduate at UBC spotted something everyone else missed.
I was looking for signs of transits, and those are when a planet passes in front of a star, it blocks a portion of the star's light, and if you're looking at measurements of the brightness, you'll see a small decrease in the brightness of the star.
Other scientists had already discovered 4 planets orbiting the distant star because they did so in a matter of days and months. But the biggest exoplanet took almost 2 Earth years to complete one orbit, and Michelle spotted 2 dips in that 4-year period. Moreover, even though it's a gas giant like Neptune, it's in that zone that's potentially habitable.
If it has a moon system, like the giant planets in our own solar system, any of those could be large enough to sustain an atmosphere and thus be able to have liquid water.
These NASA animations make it all look so simple, but screening petabytes of Kepler data is tedious stuff for most. Yet at age 22, she managed to see what others could not. Now she waits for other scientists to re-examine her discovery and for them to officially change the name from an exoplanet candidate to an exoplanet.
Students are not just working with data from space telescopes that no one else.
I have changed my shirt since then, I promise.
And that's the case with Michelle and this really exciting planet discovery.
What a day. Not only is she doing media interviews, not only is this her graduation day, this past weekend she got to meet her childhood idol as William Shatner was on campus, and in a speech he purposely talked about her exoplanet.
It was completely unexpected. I was just blown away and I could feel my face, turn red.
By the way, this planet is 3,200 light years away. What drive, Mr. Scott? Nothing the Enterprise can't handle, if only. Ted Chernecki, Global News.
And in that scene, by the way, when you saw her talking in front of this telescope, I was actually behind there trying to keep it at that angle, so that's the glamorous part of being an astrophysicist. So yes, Michelle, as I mentioned, is a huge Star Trek fan, and so it was a pleasure to introduce her to the original Captain Kirk 2 days before her graduation. And then at her graduation, UBC is giving an honorary degree to Nobel laureate Art McDonald, who won the Nobel Prize, shared the Nobel Prize in Physics in 2015. And so she got to meet a Nobel laureate 2 days later. And that just shows UBC lets me get away with a lot at convocation ceremonies up on the podium. Families and the students love it and so on. So I think and I wear something different every time. I think this is the first time I wore the wizard's robes. Now, Michelle has since started her grad studies with me at UBC, so stay tuned for more exciting discoveries. There we are in my office, which is a local tourist attraction, apparently. And I should point out that earlier this year, Michelle was named one of Forbes magazine's top 30 under 30 in science, and in fact the youngest of those at age 23. So although Farah can't be here in person, at least I've made you familiar with another, I think, inspiring woman in science. So here's just a kind of a plot of the census of planets discovered by the NASA Kepler satellite mission. On the y-axis, it's the size of the planet relative to the Earth, and I've shown Jupiter, Neptune, and Earth for scale. And then the orbital periods in days, going up to several hundred, almost 1,000 days. And if you look closely, you'll see that the new things there are like 2 years out of date, but it takes us a long time to process the data and get them out there. You want to make sure you've crossed all the T's and dotted all the I's in the analysis. Nobody wants to be, if you know the story of the boy who cried wolf, nobody wants to be the boy or girl who cried earth, and then a little while later has to say, never mind. So it takes us a while to do this. And here's another representation of the latest census. This is in terms of the sample that we have verified as of a little more than a year ago. And in this histogram, and you might, if you look at the close details, you might think, well, wow, planets that are just a little smaller than Neptune are the most common. But any investigation has selection effects and biases and so on in the sample, and we're aware of many of those, and we can correct for them. And this is what happens to that histogram if you make those corrections. And if you look closely, basically what this histogram is telling you is that 1 in 3 stars may host a planet similar in size to the Earth. Here's another version of that histogram. based on another analysis from a few years before, but it is consistent with that. My colleague Eric Pettigura presented this at a conference in Santa Barbara, a NASA meeting. And let me show you part of the paper. And so here's the kind of thing when I was watching your presentations, that's what this looked like to me, I think. The HZ here stands for habitable zone. And what you're seeing here are 5 different representations, models of the boundaries of the habitable zone, inward and outward, and including the colleague I mentioned before, Ravi Kopparapu, who actually does laboratory experiments in terms of the scattering and absorption effects of different greenhouse gases illuminated by the light from different types of stars. And I think he maybe has the best handle on the habitable zone. But no matter which one of these that you might agree with, and maybe none of them are right, almost certainly probably none of them are exactly right, but it's the last column in this table, which is the fraction of planets in the habitable zone. And basically what you're seeing here is that even conservatively, based on the latest statistics, at least 6% of small rocky planets in our Milky Way galaxy would be in the Goldilocks zones of their stars. Now, I mentioned before that our Milky Way galaxy is a city with a population of several 100 billion stars. Yeah, let's just round that down to 100 billion. 6% of 100 billion? The scientist does the arithmetic, carry the 6. 6 billion! There are potentially something like 6 billion planets that might have liquid water oceans and might satisfy some of the basic requirements for life. So the takeaway from this, and I came here on a bus from Kits, so I was looking at 24 hours in Metro on the way, and so here's my version of it, 24 nanoseconds in Astro Metro. The headline, "Our galaxy is filled with alien worlds. Scientists continue to announce discoveries of exoplanetary systems." And then the fine print, which in this case is good news, not like some legalese lurking there to bite you, "Soon someone will find evidence of life on a planet in the Goldilocks zone." And when I say soon, I would say within the next 15 to 20 years. And based on some of the kind of research that you're doing and what directions you're going with your research, you might be involved in the team. You might be leading the team that makes that announcement of possibly the first evidence of life 2.0 elsewhere, likely through the detection of the spectral signature of microbial life on an exoplanet around one of these M-dwarf stars. And I have some esteemed company in my estimate. 2 years ago, NASA Chief Scientist Ellen Stoffen said, "I think we'll have strong indications of life beyond Earth within a decade and definitive evidence within 20 or 30 years." And so we're on the same timetable at the same wavelength. Now, speaking of timetable, we're just coming up at 3:30. I have about 10 minutes more, but I could pause here. I'll leave it to my now dear friend Ed, I've known for hours and hours. If you want a few minutes more, okay. So, how will that evidence come about? So essentially, here's a picture that we don't have. I've labeled this as a star and a planet. This is a real image, but it's a star and a fainter star in a binary system. For something like the Earth, we don't have an image like this. For giant planets in supergiant orbits, we do have some images like this, but those aren't the ones where we would find life on those worlds. And so basically, we've got a few pixels of light, and we're trying to squeeze everything we can out of it, and the planet's being photobombed by the star. What kind of knowledge can we extract? Looking for biogenic signatures. So astronomy is a remote sensing science in the most remote sense of the word. And when we are seeing the light coming from a planet, even the Earth, what we're doing is seeing reflection at visible wavelengths of light from the sun or the star, scattered back to the observer, some absorption by materials on the surface and in that atmosphere and the clouds. And a warm exoplanet, and anything above 0 Kelvin is a warm object as far as an astronomer is concerned, and a physicist, emits infrared radiation which escapes to space. And so components of the atmosphere absorb that infrared and produce spectral features. And if you were to look at the Earth from space at visible wavelengths with spectrographs, and this is starting in the kind of the ultraviolet range and working up into the near infrared, and looking at different parts of the Earth, forest, deep ocean, desert, cirrus clouds. And you can see different spectra of different transmissions and features, which I've labeled here. Water vapor, one of the most dominant ones, and we should be grateful for that. Greenhouse effect and greenhouse gases are kind of a dirty word because we're concerned about a tipping point in greenhouse gases like carbon dioxide and carbon monoxide, but the greenhouse effect is a good thing. The dominant greenhouse gas in the Earth's atmosphere is water vapor. And if it weren't for the greenhouse effect due to water vapor, the average temperature on this planet would be 15 degrees below zero, about 30 degrees cooler than it is, and none of us would likely be here. So the greenhouse effect is a good thing, but in moderation. And so that's why you see so much water vapor in some of these signatures and molecular oxygen. Now here's the Earth from space at infrared wavelengths. The last chart goes up to 1.4 microns, 1.4 millionths of a meter in wavelength. This one starts at 5 microns and goes up to 25, so at a much longer range. And now you're again looking at forest and ocean and desert, now two types of clouds. And now you're seeing the ozone layer in our upper atmosphere, you're seeing the carbon dioxide, and you're seeing methane, and so on. And the best signature of life on a distant planet, at least we used to think for a long time, was methane and ozone. And in fact, instruments on the Galileo probe en route to Jupiter turned back to the Earth at the suggestion of Carl Sagan to see if those instruments could pick up signs of life on Earth. The most distinctive biosignature was that methane was far out of balance to where it should be in just chemical reactions without biology. And the reason why is because of agriculture and domestic and cattle, herds of cattle, mostly cow burps, and to a secondary degree cow farts. So that would be maybe aliens who are looking into the Earth. You would think, okay, that's our calling card, is a cow burp. That's kind of rude. But the thing is that no, aliens, unless they have any technology like ours, probably wouldn't detect life on Earth today due to methane, because although it is way out of balance and overabundance, it's still a very small concentration, 1.6 parts per million by volume. And so with the technology we have looking for signatures of life on other planets, if we were looking at a modern day Earth, we wouldn't be able to detect that methane. But, the universe has been around for 13.7 billion years based on our best estimates, and galaxies and stars and planets have been around. The Earth has been around for 4.5 billion years. What if you're looking at an older version of the Earth, or a younger version of the Earth, like in the Proterozoic era, before the Cambrian explosion? And there's a period from about 2.5 billion years ago to 500 million years ago in which there might not have been much oxygen, or if there was, it was very low levels. But there were high production of methane from marine sediments, and so that would be detectable by our current instrumentation. But you have to be careful. There was a time when everyone said, hey, if you find molecular oxygen, you found life, and that's not necessarily true. Same thing with ozone. Small planets that aren't big enough to have volcanism and plate tectonics, and if the surface is frozen so that there's no rain out of oxidants from the atmosphere, the kinds of conditions that are met on Mars today. But Mars is so small that it loses oxygen to space by what we call non-thermal processes, that energetic particles from the solar wind and ultraviolet radiation from the sun can break apart the oxygen and ozone and break it up into oxygen atoms, and then they are light enough that they can escape the weak gravity of Mars, and so that leaks off into space. And so on Mars today, that limits the oxygen mixing ratio to about 0.1%. But if Mars were just a little bit bigger, that wouldn't happen. It still wouldn't be habitable, but it would have lots of molecular oxygen in its atmosphere, but absolutely no life. So oxygen by itself isn't enough. So we got to be careful of these kind of false positives. But the trifecta, if anyone is a horse racing fan, I'm not, but I know what a trifecta is. Molecular oxygen and ozone, if you have them in conjunction with liquid water, hard to get that if you don't have life. And so that trio of signatures would be an indicator of life. Now, how are you going to find these far infrared spectra? How are you going to get them? And we don't have the instruments yet today. Here is America's big space telescope, the Hubble Space Telescope, very well known. And it's going to be retired soon and will be replaced next October, if a year from October 2018, if everything goes on schedule, by this space telescope called the JWST, which stands for James Webb Space Telescope. And here are the primary mirrors. Research telescopes don't collect light with lenses, they collect them with large mirrors. And the Hubble Space Telescope actually isn't a very large mirror by modern standards. The biggest telescopes on Earth, optical telescopes, have mirrors 10 meters across. But it's the largest that we have available in space at the moment. But this James Webb Space Telescope will have one 6.5 meters across. And the light collecting goes as the area, so it goes as the radius, the diameter squared. And notice it looks like a kind of a honeycomb. That's because we don't have any rockets big enough to send up something 6.5 meters wide. And so the mirror will unfold in space like the origami and the petals of an origami flower. At least that's what we hope. That's what's supposed to happen. Now, Hubble Space Telescope's about the size of a school bus. You could park it in a parking lot outside. You'd need a bigger parking space for James Webb, something about the size of a tennis court. And if you don't believe me, here's the full-scale mock-up of the JWST with members of the team posing outside of it on the grounds. And here's the logo, the mission patch. NASA, everybody knows, ESA, European Space Agency, CSA, not the Canadian Standards Association that you see on electrical plugs. Canadian Space Agency. Canada is a major partner in this exciting project. And JWST is going to use its instrumentation to look in the far infrared part of the spectrum. It's going to investigate places where planets are forming even as I speak. Going to figure out how planets form, how are habitable zones established. It will look at objects in our own solar system to find analogs, to interpret these other distant sources, and it will actually look at planets that are passing in front of their stars, and we call it transiting, and will look for the signatures in the atmosphere as the light from the star is filtered by the atmosphere, the thin atmosphere of the planet around it. Now, when we're talking about life, we tend to think of it on the surface of a planet, and we do talk about the thin atmosphere. And here's a picture taken from the International Space Station, seeing light scattered from the sun off in the distance, off our thin atmosphere. When I say thin, how thin? Well, if you were to shrink the Earth down to the size of an apple, our atmosphere would be thinner than the skin of that apple. And it's within that thin layer that we have life on Earth and where we would be looking for life on other worlds. But it turns out that's not the whole story. When we think about worlds, we think of planets, and when we think about life, we tend to think of planets. I've already shown you a case of Michel's discovery of warm Neptune, that if it has large moons around it, that those would be places to find life. But even within our solar system, there are places. You know, we've been thinking about Mars, we've been kind of obsessed with Mars for a long time, but that's not the full story. Here is just a partial list of the other places in our solar system where we strongly suspect that there might be life. And the life's not on the surface if it's there. And these are deep habitats. These are icy worlds far outside of the Goldilocks zone, too cold to have liquid water on the surface, but beneath their icy crusts we have evidence of deep oceans. And some of those oceans would exist around 3 of the large moons of Jupiter that I talked about before, the Galilean moons, and 1 tiny moon of Saturn called Enceladus. Europa, which is a planet that's roughly about the size of our moon, or a moon that's roughly about the size of the Earth's moon, it's believed to have an ocean several hundred kilometers deep beneath it. And so we think of ourselves as the king of the solar system, queen of the solar system in terms of water, but if all measurements are right, this tiny moon, Europa, contains twice as much water as we have on Earth. And so this is a new perspective for life. And the largest moon around Saturn, called Titan, appropriately, not only has a subsurface ocean, maybe there's life there, although there's some worries about that, but its surface has liquid methane, It's got liquid methane lakes and liquid methane rivers and liquid methane rain. So it's an environment, it's weather, except that liquid methane replaces water. And remember when I talked about water as a solvent and transport medium and ammonia was second best? Well, there are now serious considerations, we're looking into it, that on Titan, that life might be able to exist using liquid methane at like cryogenic temperatures to be its solvent and transport medium. And we already know that Titan has all of the raw materials in terms of organics to be able to assemble life. And so this is the kind of thing that would have had me laughed out of a room 10 years ago if I talked about this. And even science fiction writers would have laughed. I mean, George Lucas would have said, "Oh, that's ridiculous," and he's the guy that came up with Jar Jar Binks. And so my message is the fact that that's even more options for life in our galaxy and our universe, not just in the Goldilocks zone, but deep habitat and methane worlds. So going back to the newspapers, more Goldilocks worlds will be found. Planets in the habitable zone satisfy at least one requirement for life as we know it. Again, I invite you to look at the fine print. Life may also thrive under the surfaces of icy moons in deep habitat oceans. And so with that, I don't think there's going to be formal time for questions here, because I know you have a barbecue waiting for you, but I will stick around for as long as I can. I have to be at the H.R. McMillan Space Center, or planetarium, at 6:30 for an event tonight mixing art and science. It's called Eclipse, Total Alignment, with dance and art and poetry and music and science. But I have enough time to hang out here, and if anyone who wants to approach me one-on-one, don't be afraid, even though these journalists are nervous about talking to me after these experiences on Skype and TV interviews. But that's my own. I wasn't at a club, by the way, in the upper left and lower right. I travel sometimes with my own portable laser light show, so I thought that would be a good backdrop from my hotel rooms in various cities to connect with the media. Anyway, thank you very much for your patience.
Don't go away.
Oh.
Don't go away. So there is something in here. Oh. There was a plaque, but it says ferret. So I don't think you want that one.
Well, I don't know.
But that's for you.
Oh,
right?
thank you.
I think they're taking pictures.
Oh.
Hi. You're good. Okay, and don't go away. Okay, so thank you so much for this last minute, really appreciate it in the middle of all this media about the solar eclipse. We're really thrilled that you came. So thank you very much.